Welcome, fellow dreamer. You have found a place of calm and rest. This evening, we're going to explore one of science's most elegant mysteries, the story of DNA, a graceful spiral that holds the secret instructions for all living things, written in just 4 simple letters. Accompanied by soothing music and abstract imagery, we'll be uncovering little-known wonders and fascinating stories that gently feed your curiosity as you drift towards sleep. If you enjoy these calm adventures with us, don't be shy. Like, subscribe, and leave a comment to help other smart sleepers dream too. And now, take a deep breath, let your body sink into comfort, and allow your mind to rest and your eyes to close as the tale unfolds before you. The Mediterranean sun cast long shadows across the marble steps of the Academy in Athens, where philosophers gathered to debate the mysteries of existence. Among their many questions, one puzzle seemed particularly persistent. Why do children resemble their parents? Why does a farmer's daughter have her mother's eyes, or a merchant's son inherit his father's stature? These questions, simple enough for any child to ask, would occupy humanity's greatest minds for more than 2,000 years. Aristotle, walking among his students in the Lyceum gardens, proposed that new life arose from a mixture of materials provided by both parents. He imagined that the father contributed form and motion, while the mother provided the matter from which the child developed. It was an elegant theory for its time, grounded in the philosophical principles that governed Greek thought. Yet, like many ancient explanations, it described what people could observe without revealing the mechanism hidden beneath. The Roman naturalist Pliny the Elder, compiling his extensive natural history, recorded countless observations about inheritance in plants and animals. He noted how certain traits seemed to skip generations, appearing in a grandchild though absent in the parent. He documented the careful breeding practices of farmers who selected their best crops and livestock, somehow knowing through experience what philosophy could not yet explain. These practical agriculturists had discovered something real, something that worked, even if they couldn't say why. As centuries passed and the classical world gave way to medieval scholarship, new theories emerged from careful observers. Persian and Arab scholars made detailed studies of inheritance, examining family resemblances and the transmission of physical characteristics. They noted that traits could blend, but sometimes appeared unmixed, as if following rules no one had yet written down. In the libraries of Baghdad and Cordoba, manuscripts accumulated observations that would have delighted the Greek philosophers of old, yet the fundamental question remained unanswered. European scholars of the Renaissance, rediscovering classical texts and adding their own observations, continued to wrestle with these mysteries. They watched dogs bred for hunting produce puppies with similar instincts. They observed how apple trees, grown from carefully selected seeds, might yield fruit resembling the parent tree, or sometimes something entirely different. The natural world seemed to follow patterns, but patterns that shifted and changed in ways that defied simple explanation. Some theorists proposed that tiny, fully formed creatures existed inside seeds or eggs, simply waiting to unfold and grow larger. This theory, called preformation, suggested that all future generations had been created at the beginning of time, nested inside one another like an infinite set of wooden dolls. It was a tidy explanation that avoided the messy question of how something new could arise from formless matter. Others championed epigenesis, the idea that organisms developed gradually from undifferentiated material, somehow guided by forces yet unknown. The debate between these camps grew heated, with experiments and observations marshaled by both sides. In the botanical gardens of Europe, naturalists catalogued the extraordinary diversity of plant life and began attempting systematic breeding experiments. They noticed that crossing different varieties sometimes produced offspring that resembled one parent more than the other. Other times, the offspring showed a mixture of characteristics or even traits that seemed entirely new. The Austrian botanist Joseph Gottlieb Kohlreuter, working in the 18th century, performed thousands of careful crosses between plant species, documenting the results with meticulous attention. His work revealed that heredity followed some kind of order, but the underlying principles remained frustratingly out of reach. the French naturalist Jean-Baptiste Lamarck proposed that organisms could pass acquired characteristics to their offspring. A giraffe stretching to reach high leaves might, through this effort, develop a longer neck, and this acquired trait would then appear in its descendants. The theory felt intuitively correct to many observers. After all, didn't the blacksmith's children seem stronger, as if inheriting the muscles their father had built through years of labor? The idea that use or disuse of body parts could be inherited offered an explanation for how species might change over time, adapting to their environments across generations. When Charles Darwin published his revolutionary theory of evolution by natural selection, he provided a compelling explanation for how species changed over time, but he couldn't explain the mechanism of inheritance itself. He knew that variation existed in populations, and that parents passed traits to offspring, but the question of how this happened remained a mystery. Darwin himself proposed a theory involving particles called gemmules that traveled from all parts of the body to the reproductive organs, carrying information about each organ's characteristics. It was an ingenious guess, but it was wrong. As the 19th century progressed, scientists made increasingly sophisticated observations of reproduction in animals and plants. They examined pollen and seeds under improved microscopes, watched the development of embryos, and documented the outcomes of countless breeding experiments. The conviction grew that inheritance must follow natural laws, like gravity or magnetism, laws that could be discovered through patient observation and experiment. Yet those laws remained hidden, encoded in mechanisms too small to see and too complex to guess. Farmers and breeders, meanwhile, continued their practical work, developing new varieties of crops and livestock through selection. They knew from experience that breeding the best individuals produced better offspring, though the results were never entirely predictable. Sometimes desirable traits seemed to disappear, only to resurface in later generations. Sometimes weak or unusual offspring appeared from the strongest parents. The patterns were there, tantalizingly consistent, yet frustratingly incomplete. By the middle of the 19th century, the stage was set for a breakthrough. Microscopes were improving, allowing scientists to peer deeper into the structure of living tissues. The scientific method was becoming more rigorous, with controlled experiments replacing speculation. Most importantly, researchers were beginning to ask more precise questions, looking for mathematical patterns rather than philosophical principles. Somewhere in Europe, the right person in the right place with the right approach might finally glimpse the hidden architecture of heredity. That person would not be a university professor at a great research institution. He would not have access to the finest laboratories or the most advanced equipment. Instead, in a quiet monastery garden in a corner of the Austrian Empire, a teacher with a passion for understanding nature's patterns was about to begin experiments that would, decades later, transform humanity's understanding of life itself. In the quiet town of Biarono, nestled in what is now the Czech Republic, a man in simple monastic robes walked through his garden on a spring morning in the 1850s. The garden behind the Augustinian monastery was not large, but it was carefully tended, and this particular monk had plans for it that would reach far beyond its walls. His name was Gregor Mendel, and though he could not have known it then, he was about to unlock one of nature's most fundamental secrets. Mendel was a curious soul, born into a farming family, where he had learned from childhood to observe the patterns of nature. He had noticed, as farmers had for generations, that certain traits seemed to pass from parent plants to their offspring, but the process appeared frustratingly unpredictable. Sometimes a trait would vanish for a generation, only to reappear mysteriously in the next. It was as if nature were playing by rules that no one had yet discovered. The monastery had sent him to study physics and mathematics at the University of Vienna, and it was there that something shifted in his thinking. He learned that natural phenomena could be measured, counted, and analyzed with mathematical precision. When he returned to Biarono, he brought with him an unusual idea. Perhaps inheritance, too, followed mathematical laws that could be revealed through careful experimentation. He chose his experimental subject with practical wisdom. Pea plants were easy to grow, produced many offspring in a short time, and came in varieties with clearly distinguishable traits. Some plants grew tall, while others remained short. Some produced smooth seeds, while others had wrinkled ones. Some bore yellow seeds, others green. Most importantly, pea plants could be controlled with precision. Their flowers could be carefully opened and pollinated by hand, allowing Mendel to act as matchmaker, deciding exactly which plants would contribute to the next generation. In his garden plot, Mendel began his experiments with the patience that would characterize all his work. He started by ensuring he had true breeding lines, plants that consistently produced offspring identical to themselves, generation after generation. This preliminary work alone took time, but Mendel understood that good science required a solid foundation. Only when he was certain his starting lines were pure did he begin the crosses that would reveal nature's patterns. He took pollen from tall plants and carefully transferred it to the flowers of short plants. He protected these flowers from random pollination by insects or wind, ensuring that only his chosen parents contributed to the seeds that formed. Then he waited, with the particular patience of someone who tends gardens, for the plants to grow and reveal their secrets. When the first generation of hybrid plants matured, they were all tall, every single one. The shortness had seemingly vanished, as if the tall characteristic had completely overwhelmed it. This matched what many plant breeders had observed before, the apparent dominance of certain traits, but Mendel did not stop there. Where others had concluded their observations, he allowed these hybrid plants to self-pollinate and collected their seeds. The following spring, he planted these second-generation seeds, and as the plants grew, something remarkable appeared. The shortness had returned. Among roughly every 4 plants, 3 were tall and 1 was short. Mendel counted carefully, plant by plant, trait by trait. The ratio appeared again and again, approximately 3 to 1, with a consistency that could not be coincidence. He repeated the experiment with other traits. When he crossed plants with smooth seeds and wrinkled seeds, the first generation was all smooth, but the second generation showed that familiar ratio, 3 smooth to 1 wrinkled. Yellow seeds crossed with green produced all yellow in the first generation, then 3 yellow to 1 green in the second. The pattern held across different characteristics, a mathematical signature, underlying inheritance. Through years of careful work, counting thousands upon thousands of plants, Mendel developed his explanation. He proposed that traits were controlled by paired factors, with one factor inherited from each parent. Some factors were dominant, expressing themselves even when paired with a different version, while others were recessive, expressing themselves only when 2 copies came together. When parents formed reproductive cells, these paired factors separated, with each cell receiving only one. When egg and pollen united, the pairs were restored in the offspring. It was an elegant theory, supported by mountains of careful data. Mendel presented his findings to the Natural History Society of Brno in 1865, speaking to a small audience in that provincial city. He published his results the following year in the Society's journal, explaining his experiments and the mathematical laws he had uncovered. The paper was clear, methodical, and revolutionary. And then, almost nothing happened. A few copies of the journal made their way to libraries and scientific institutions across Europe. Perhaps some botanists glanced at the paper, but if they did, they failed to grasp its significance. Mendel sent copies to prominent scientists, but received little response. His work was too mathematical for most biologists of the time, too focused on an obscure crop plant, too far ahead of the conceptual framework that would be needed to appreciate it. Mendel continued tending his garden and pursuing other experiments, but administrative duties increasingly occupied his time as he rose in the monastery's hierarchy. Eventually, he became abbot, and the responsibilities of leadership drew him away from his pea plaints. His paper remained on dusty library shelves, cited rarely if at all, its importance unrecognized. When Mendel died in 1884, he left behind the garden where he had discovered the fundamental laws of inheritance, laws that the scientific world had not yet realized it needed. His work lay waiting, like seeds in winter soil, ready to germinate when the time was right. That time would come, though Mendel himself would never see the spring that his discoveries would bring to the science of heredity. The patterns he had so carefully documented would eventually be understood as the foundation of an entirely new field of biology. But first, other pieces of the puzzle needed to fall into place. The microscope changed everything. In those last decades of the 19th century, as gaslight gave way to electric illumination in the laboratories of Europe and America, a new world revealed itself beneath glass lenses. Scientists peering through eyepieces discovered that all living things, whether plant or animal, were composed of tiny chambers that an early observer had called cells, after the small rooms monks occupied in monasteries. These cells were not merely simple sacs of fluid. As microscope makers refined their instruments, grinding better lenses, and devising more sophisticated illumination techniques, the interior landscape of the cell came into focus like a city emerging from morning fog. There were structures within structures, each with its own character and perhaps its own purpose. The cell nucleus, a darker sphere within the cell's boundaries, drew particular attention. It seemed to be the command center, the heart of the cell's operations, though what exactly it commanded remained mysterious. The act of observing cells required patience and stillness. A scientist might spend hours adjusting mirrors to catch the light just so, preparing thin slices of tissue on glass slides, applying dyes that would stain some cellular components while leaving others transparent. The dyes themselves were often discoveries of accident, borrowed from the textile industry, where chemists had learned to color fabrics in brilliant hues. These same compounds, it turned out, had an affinity for certain parts of the cell, making visible what had been invisible. In the 1870s and 1880s, several researchers noticed something remarkable occurring when cells divided. Walter Fleming, working in Prague and later in Kiel, watched with fascination as thread-like structures appeared in the nucleus of dividing cells. These threads seemed to materialize from the seemingly uniform substance of the nucleus, growing darker as they absorbed the dyes he applied. Fleming called them chromosomes, from the Greek words for colored bodies, and he traced their behavior through the complete process of cell division, which he termed mitosis. The dance of the chromosomes was intricate and precise. As a cell prepared to divide, these thread-like structures became visible, each one duplicating itself. The duplicated chromosomes lined up along the cell's equator, like dancers taking their positions. Then, as if responding to some unheard music, they separated, with one copy moving to each end of the dividing cell. When the cell finally split into two daughter cells, each received an identical set of chromosomes. The precision of this distribution suggested purpose, though what exactly was being distributed remained unclear. Eduard Strasberger observed similar processes in plant cells, confirming that this was not a peculiarity of animal tissue, but rather a fundamental feature of life itself. The consistency of the process across different organisms hinted at something important. Nature does not usually preserve mechanisms unless they serve essential functions. But what function did these chromosomes serve? Other scientists added pieces to the puzzle. Oscar Hertwig, studying sea urchin eggs, watched as a sperm cell fused with an egg cell during fertilization. He observed that both sperm and egg contributed a nucleus to the fertilized egg, and within those nuclei were chromosomes. The fertilized egg thus contained chromosomes from both parents. This observation connected the visible structures under the microscope with the patterns of inheritance that Mendel had described in his pea plants, though few scientists had yet recognized Mendel's work. Wilhelm Roux proposed that chromosomes might not be simple threads, but rather composed of distinct units arranged along their length, like beads on a string. Each unit might carry different hereditary information. This was speculation, certainly, but it was speculation grounded in careful observation. The way chromosomes split lengthwise during cell division suggested they had an internal structure, something more than mere threads of uniform material. August Weismann, pondering these observations from his laboratory in Freiburg, developed a theory that distinguished between body cells and reproductive cells. He proposed that only reproductive cells, the eggs and sperm, passed hereditary information to the next generation. The chromosomes in these cells must therefore be the carriers of inheritance. Body cells might change during an organism's lifetime. Muscles might strengthen or skin might tan, but these changes were not inherited because they did not alter the chromosomes in reproductive cells. But this insight helped explain why children did not inherit traits their parents had acquired through use or habit, a question that had puzzled thinkers for generations. The connection between chromosomes and inheritance was becoming clearer, though still not proven. Theodore Boveri, working with sea urchin eggs in Naples, performed elegant experiments that suggested individual chromosomes carried different hereditary information. He created abnormal embryos with unusual numbers of chromosomes and observed that they developed abnormally, each in characteristic ways depending on which chromosomes were missing or duplicated. This suggested that chromosomes were not all identical carriers of the same information, but rather each one contributed something unique to the organism's development. In laboratories scattered across Europe and America, in buildings that smelled of chemical fixatives and were lit by the warm glow of gas mantles or the harsh light of early electric bulbs, scientists were sketching what they saw through their microscopes. Their notebooks filled with drawings of cells in various stages of division, chromosomes rendered in careful detail. Some of these scientists were professors at universities. Others were independent researchers pursuing questions that fascinated them. They corresponded with each other, sending letters that took weeks to cross oceans, sharing observations and debating interpretations. The technology of the microscope itself continued to improve. Better lenses reduced the colorful halos that plagued earlier instruments. Mechanical stages allowed precise movement of slides. Oil immersion techniques increased magnification and clarity. Each improvement revealed new details, answered old questions, and raised new ones. By the final years of the 19th century, the cell theory was firmly established, and chromosomes were recognized as consistent features of cell division and reproduction. The idea that these structures might carry hereditary information was gaining support, though it remained a hypothesis rather than proven fact. The mechanisms were still obscure. How could these visible threads under the microscope relate to the mathematical patterns Mendel had found in his garden? How did chromosomes encode the instructions for building an organism? These questions hung in the air of laboratories as the old century gave way to the new. The tools for answering them were being assembled, the observations carefully recorded, the stage set for the next act in this unfolding story of discovery. As the 19th century drew to a close, Mendel's carefully documented experiments lay forgotten in the pages of an obscure scientific journal. The Augustinian monk had passed away in 1884, his revolutionary insights into inheritance unrecognized by the scientific community. His work had been published, catalogued, and then essentially abandoned, gathering dust in libraries across Europe while the world of biology moved forward without him. But science has a peculiar way of correcting itself, of finding truths that have been overlooked. Sometimes discovery happens not once, but multiple times, as if nature itself insists that certain secrets must eventually come to light. The year 1900 arrived with the turning of a new century, bringing with it a sense of possibility and change. In 3 different locations across Europe, 3 botanists were conducting their own experiments with plant breeding, each working independently, unaware of the others. Each man was asking similar questions about how traits pass from one generation to the next, and each was about to make a remarkable discovery. In Amsterdam, Hugo de Vries was working with evening primrose plants in his garden. The Dutch botanist had spent years observing how characteristics appeared and disappeared across generations. As he analyzed his data, mathematical patterns began to emerge. Certain traits seemed to follow predictable rules, appearing in specific ratios among offspring. De Vries was meticulous in his record-keeping, counting plants with different characteristics and noting the proportions with care. As he prepared to publish his findings, de Vries did what any good scientist should do. He searched through earlier scientific literature to see if anyone had worked on similar problems, and there, in the dusty pages of a journal from 1866, he found Mendel's paper. The monk's work, conducted more than 3 decades earlier, described the same patterns de Vries had just discovered. The ratios matched, the principles aligned, Mendel had been there first. Meanwhile, in Germany, Karl Correns was conducting his own breeding experiments. Correns worked with peas and maize, carefully cross-pollinating plants and tracking traits through multiple generations. He too noticed the mathematical regularities in how characteristics were inherited. Like de Vries, he observed that traits didn't blend but remained distinct, appearing and disappearing according to predictable rules. Correns also searched the scientific literature, and he too found Mendel's paper. But for Correns, the discovery carried a particular resonance. He had studied under Carl von Nägeli, the same botanist to whom Mendel had sent his work years earlier, seeking recognition and support. Nägeli had dismissed Mendel's findings, considering them unremarkable. Now his own student was rediscovering the same principles, recognizing their profound importance. In Vienna, a third botanist named Erich von Charmack was conducting similar experiments, also with peas. The young Austrian researcher was observing the same inheritance patterns, the same mathematical ratios, appearing generation after generation. When he too came across Mendel's forgotten paper, he realized that the fundamental laws of heredity had already been described, waiting patiently to be recognized. The spring of 1900 saw all three scientists rushing to publish their findings. Each acknowledged Mendel's priority, recognizing that the monk had discovered these principles first, but their independent confirmation gave Mendel's work something it had lacked in his own lifetime: validation. When three different researchers, working with different plants in different locations, all arrive at the same conclusions, the scientific community takes notice. The timing was perfect. The biology of 1900 was ready for Mendel in a way that the biology of 1866 had not been. The intervening decades had brought the discovery of chromosomes, the observation of cell division, and a growing sense that heredity must have a physical basis in cellular structures. Mendel's abstract factors, his units of inheritance, suddenly seemed to fit with what microscopes were revealing about cells. The rediscovery sparked immediate interest. Scientists across Europe and America began conducting their own breeding experiments, confirming and extending Mendel's principles. The word genetics had not yet been coined, but the field was being born. Mendel's laws of inheritance, the patterns of dominance and recessiveness, the independence of different traits, all became foundation stones for a new science. William Bateson, an English biologist, became one of Mendel's most enthusiastic champions. He translated Mendel's paper into English and promoted the monk's work vigorously. In 1906, Bateson coined the term genetics to describe this new field devoted to understanding heredity and variation. The science finally had a name. The rediscovery also raised new questions. If Mendel's factors controlled inheritance, what were these factors made of? Where exactly were they located in cells? How did they create the visible characteristics of organisms? The patterns were clear, but the mechanism remained mysterious. Scientists knew that something was being passed from parent to offspring according to mathematical rules, but the physical nature of that something was still unknown. Some researchers began to suspect that chromosomes, those thread-like structures visible during cell division, might be involved. The behavior of chromosomes during the formation of reproductive cells seemed to match the behavior of Mendel's factors. Chromosomes came in pairs, just as Mendel's factors did. They separated during the formation of egg and sperm cells, just as his factors separated. The parallel was suggestive, though not yet proven. The year 1900 marked a turning point. What had been isolated observations and scattered theories began to coalesce into a coherent framework. Mendel's laws provided a mathematical foundation. Chromosomes provided a potential physical basis. Together, they pointed toward a unified understanding of inheritance. As news of the rediscovery spread through universities and research institutions, a new generation of scientists became excited about studying heredity. The question of how traits pass from parent to offspring, which had puzzled humanity for millennia, suddenly seemed approachable through careful experimentation and mathematical analysis. The forgotten monk's work had been rescued from obscurity, and a new science was emerging into the light. The early years of the 20th century brought electricity to city streets, automobiles to dusty roads, and a new urgency to the study of inheritance. With Mendel's laws rediscovered and confirmed, scientists across Europe and America turned their attention to a pressing question: where exactly did these mysterious factors of inheritance reside? The abstract patterns Mendel had observed in his pea plants clearly followed mathematical rules, but what physical structures carried these instructions from parent to offspring? On the 17th floor of Schermerhorn Hall at Columbia University in New York City, a narrow room measuring only 16 by 23 feet would become the unlikely birthplace of revolutionary discoveries. This cramped space, soon to be affectionately known as the Fly Room, housed an eccentric collection of researchers led by Thomas Hunt Morgan, a biologist from Kentucky with a skeptical mind and an appetite for evidence. Morgan had initially doubted Mendel's theories, believing that inheritance might be far more complicated than simple dominant and recessive factors could explain. But he was a man devoted to experimentation above all else, and he needed an organism that could help him test these ideas thoroughly. The creature he chose seemed almost comically small for such grand ambitions: Drosophila melanogaster, the common fruit fly. These tiny insects, barely 3 millimeters long, possessed qualities that made them perfect for genetic research. They reproduced quickly, with a new generation emerging every 2 weeks. They required minimal space and could survive happily in glass milk bottles, eating nothing more than mashed bananas mixed with a bit of agar. A single pair could produce hundreds of offspring, providing abundant data for statistical analysis. And perhaps most importantly, they had only 4 pairs of chromosomes, making them simpler to study than organisms with more complex genetic architecture. The Fly Room became a peculiar world unto itself. Bottles containing thousands of flies lined the shelves and covered every available surface. The sweet smell of fermenting bananas permeated the air. Researchers worked shoulder to shoulder at narrow benches, peering through hand lenses at flies anesthetized with ether, counting the subtle variations in eye color, wing shape, and body patterns. The windows remained closed despite the summer heat, lest a breeze release the precious experimental subjects into the Manhattan air. Morgan's team included brilliant young scientists who would themselves become legendary figures in genetics. Alfred Sturtevant arrived as an undergraduate and would make groundbreaking discoveries before finishing his degree. Calvin Bridges brought mathematical precision to their observations. Hermann Müller contributed theoretical insights that pushed their thinking in new directions. Together, they created an atmosphere of intense collaboration, where ideas flowed freely and discoveries emerged from collective effort. The breakthrough came in 1910, when Morgan noticed something unusual. Among thousands of flies with normal red eyes, a single male appeared with eyes of pure white. This wasn't a subtle variation, it was a dramatic difference that bred true when the white-eyed male was crossed with red-eyed females. The inheritance pattern that emerged puzzled Morgan at first. All the offspring in the first generation had red eyes, suggesting red was dominant, just as Mendel's laws would predict. But in the second generation, something unexpected appeared. Only males displayed white eyes. The trait seemed somehow connected to sex. Morgan realized he was observing something profound. The white eye trait was inherited along with maleness, suggesting it must be physically located on the same chromosome that determined sex. This was the first clear evidence that specific traits were carried on specific chromosomes. The abstract factors Mendel had proposed weren't floating freely in cells. They occupied particular physical locations on these thread-like structures, visible under microscopes. This discovery opened floodgates of understanding. The Fly Room team began identifying dozens of different mutations, each affecting some aspect of the fly's appearance or behavior. Some mutations changed body color from gray to yellow or black. Others altered wing shape, creating miniature wings or wings with notched edges. Still others affected eye color in various ways, producing vermilion, sepia, or brown eyes instead of the normal red. As more mutations were identified, patterns emerged. Certain traits tended to be inherited together, traveling as groups through generations. Sturtevant, working late one night, suddenly grasped what this meant. If genes were arranged in linear order along chromosomes like beads on a string, then genes located close together would usually be inherited as a unit, while genes far apart might separate during the shuffling that occurred in reproduction. The frequency with which traits separated could reveal their physical distances from each other. In a single remarkable evening, 19-year-old Sturtevant created the first genetic map, showing the relative positions of 5 genes along a single chromosome. He worked through the night, neglecting his homework assignment to pursue this unexpected insight. His map, drawn on a piece of scratch paper, represented a conceptual leap of stunning importance. Genes weren't just abstract factors, they occupied specific, mappable locations on chromosomes. The architecture of inheritance could be charted and understood. The fly room's productivity seemed inexhaustible. Year after year, new mutations appeared, new patterns emerged, and the maps grew more detailed. Morgan's initial skepticism transformed into conviction as evidence accumulated beyond any reasonable doubt. Genes existed as physical entities arranged along chromosomes. They could mutate, creating new variations. They could be mapped relative to each other, and the principles governing fruit fly inheritance appeared to apply across the living world. The tiny fruit fly, so insignificant it might escape notice at a summer picnic, had revealed truths about the mechanism of heredity that applied to all organisms with chromosomes. The same principles operating in Drosophila operated in plants, in mice, in humans. The universality of these findings suggested something even more profound. All life shared a common genetic logic, a fundamental unity beneath its surface diversity. By the time Morgan received the Nobel Prize in 1933, the fly room had established genetics as a rigorous experimental science with predictive power. What had begun as patterns in pea plants had become a detailed understanding of how chromosomes carried discrete units of inheritance in specific mappable locations. Yet even with all these advances, a crucial question remained unanswered. The fly room had proven where genes were and how they behaved, but not what they were made of. That mystery would require different tools and different minds to unravel. By the late 1920s, scientists had mapped genes along chromosomes, like towns along a railroad line. They knew that these mysterious units of heredity controlled traits, from eye color in fruit flies to flower patterns in plants. But beneath this growing understanding lay a profound question that nobody could answer. What exactly were genes made of? The question hung in laboratories across the world like morning mist, tantalizingly close yet somehow beyond reach. Scientists could see the effects of genes, could track their inheritance through generations, could even map their positions. But the fundamental chemical nature of heredity remained hidden. It was rather like knowing that letters exist and understanding their arrangement matters, yet having no idea whether they were made of ink, paint, or something else entirely. The debate that emerged during this period centered on 2 very different kinds of molecules found in cells. On one side stood proteins, those versatile and complex substances that seemed capable of almost anything. On the other side were nucleic acids, relatively simple molecules that few scientists considered promising candidates for carrying the instructions of life. Proteins appeared to be the obvious answer. These remarkable molecules were known to be incredibly diverse, built from 20 different building blocks called amino acids. The variety seemed endless. Some proteins formed structures like hair and fingernails. Others acted as enzymes, speeding up chemical reactions. Still others transported oxygen through blood or fought off infections. Surely, scientists reasoned, such versatility made proteins the perfect molecules for encoding the vast diversity of hereditary information. The sheer complexity of proteins seemed to match the complexity of inheritance itself. When researchers looked at the differences between species, between individuals, between traits, they saw variations that appeared to require an equally sophisticated molecular foundation. Proteins fit this expectation beautifully. They could fold into intricate shapes, perform countless functions, and existed in seemingly infinite varieties. Nucleic acids, by contrast, seemed disappointingly simple. These molecules had been discovered in the nuclei of cells back in the 1860s by a Swiss physician named Friedrich Miescher. He had been studying the chemistry of white blood cells collected from discarded surgical bandages an unglamorous beginning for such an important molecule. Meiser had isolated a substance he called nuclein, rich in phosphorus, and unlike any protein, in the decades that followed, chemists had worked out the basic structure of nucleic acids. They found these molecules were built from only 4 different building blocks, called nucleotides. Each nucleotide contained a sugar molecule, a phosphate group, and one of 4 bases. In DNA, the 4 bases were adenine, guanine, cytosine, and thymine, just 4 letters in the alphabet of heredity, compared to the 20 amino acids that made up proteins. To most scientists of the 1920s and 1930s, this simplicity seemed fatal to any possibility that nucleic acids could be genetic material. How could a molecule with only 4 components encode the instructions for building an entire organism? It was rather like asking whether a language with only 4 letters could express the full range of human thought and experience. The idea seemed implausible. Moreover, early analysis had suggested that nucleic acids might be monotonously regular in structure. Some researchers proposed that the 4 bases appeared in equal amounts, repeating in a predictable pattern. If this were true, nucleic acids would be even less capable of carrying complex information. A message written with 4 letters repeating in the same order over and over could hardly specify the subtle differences between species or the variations that made each individual unique. Proteins, meanwhile, seemed to shine brighter the more scientists learned about them. Researchers were discovering that proteins could be incredibly specific in their functions. A particular enzyme might speed up one chemical reaction but ignore hundreds of others. Antibodies could distinguish between nearly identical molecules. This specificity suggested a capacity for encoding precise information. The scientific community settled into a comfortable consensus. Genes must be made of proteins, perhaps with nucleic acids playing some supporting role. Nobody quite knew what nucleic acids did, but they seemed to be merely structural components, perhaps a scaffolding that held the important protein genes in place. The genetic material, the carrier of hereditary instructions, had to be protein. Yet even as this view took hold, a few quiet voices expressed doubt. The certainty about proteins rested more on their apparent suitability than on direct evidence. Nobody had actually proven that proteins were genetic material. Nobody had demonstrated how proteins could copy themselves with the precision that heredity required. The assumption, however reasonable it seemed, remained just that, an assumption. And there were curious facts that didn't quite fit. Chromosomes, those thread-like structures that clearly carried hereditary information, contained both proteins and nucleic acids in roughly equal amounts. If proteins were the genes, why did nucleic acids feature so prominently in chromosomes? Why were they there at all? Some researchers noted that the amount of DNA in cells showed interesting patterns. Body cells contained twice as much DNA as reproductive cells, which made sense if DNA played some role in heredity. The amount of DNA was also fairly consistent within a species, while protein content varied considerably depending on cell type and function. These observations didn't prove anything, but they hinted that perhaps nucleic acids deserved more attention than they were receiving. The question hung there, tantalizingly unanswered. As the 1930s progressed, scientists had the tools to study the chemistry of cells in ever greater detail. They had microscopes to observe chromosomes and their behavior. They had decades of genetic experiments showing how traits passed from parents to offspring. Yet the fundamental question, what are genes made of, remained a mystery, waiting for someone to devise the right experiment to answer it. In laboratories scattered across continents, researchers continued their work, most following the well-worn path toward proteins, a few beginning to wonder if the answer might lie elsewhere. The stage was set for experiments that would finally reveal the chemical nature of heredity, though few could have predicted how surprising the answer would turn out to be. The molecule that carried the secret of heredity remained elusive as scientists entered the late 1920s. Laboratories around the world buzzed with theories and experiments, each researcher hoping to be the one who would finally identify the substance that passed traits from one generation to the next. The answer would come from an unexpected place, from a quiet medical officer studying deadly bacteria in a small laboratory in England. Frederick Griffith was not searching for fame or fundamental truths about heredity. He was simply trying to understand pneumonia, the lung infection that killed so many people in the years following the First World War. In his laboratory at the Ministry of Health in London, Griffith worked with 2 strains of pneumococcus bacteria. One strain wore a smooth, glistening coat and caused deadly disease when injected into mice. The other strain appeared rough and harmless, unable to cause infection. It seemed straightforward enough, but Griffith's careful nature led him to try combinations of these bacterial strains that would reveal something extraordinary. In 1928, Griffith performed an experiment that seemed simple but would echo through the decades. He heated the smooth, deadly bacteria until they died, then injected these dead cells into mice. As expected, the mice survived. Dead bacteria could not cause disease. But then Griffith tried something unusual. He mixed the dead smooth bacteria with living rough bacteria, neither of which should have been harmful in their current states. He injected this mixture into mice, expecting them to survive. Instead, the mice died. When Griffith examined their bodies, he found living smooth bacteria, the deadly type with the glistening coat. Something had transformed the harmless bacteria into killers. Some substance from the dead smooth cells had entered the living rough cells and changed them permanently. Even more remarkable, when these transformed bacteria reproduced, their offspring retained the smooth, deadly characteristics. The change was heritable, passed from one generation to the next. Griffith called this mysterious phenomenon transformation, though he had no idea what substance was responsible for this remarkable change. The scientific community received Griffith's discovery with puzzlement and interest. The implications seemed enormous, yet the mechanism remained hidden. What molecule could carry information from dead cells to living ones and permanently alter their nature? Griffith himself was cautious, publishing his findings without grand claims. Tragically, he would not live to see the full impact of his work. Frederick Griffith died during the German bombing of London in 1941. His modest laboratory destroyed, his careful experiments remembered by those who would build upon them. The question Griffith left behind captured the attention of Oswald Avery, a physician and researcher at the Rockefeller Institute in New York. Avery was a meticulous scientist, soft-spoken and methodical, known for his extraordinarily careful approach to experiments. He had spent years studying pneumococcus bacteria himself, and Griffith's transformation experiments fascinated him. If something in dead bacteria could transform living ones, identifying that something would reveal the very substance of heredity. For Avery and his colleagues, Colin MacLeod and Macklin McCarty, began a painstaking series of experiments that would span more than a decade. Their approach was elegant in its logic. They would prepare extracts from the smooth bacteria, then systematically destroy different types of molecules in these extracts. Whichever molecule they could not destroy without eliminating the transforming ability must be the one carrying the genetic information. They worked through the possibilities with careful patience. They treated their bacterial extracts with enzymes that destroyed proteins. The transformation still occurred. They destroyed lipids and carbohydrates. Still, the harmless bacteria became deadly when exposed to the extract. The scientific consensus of the time held that genes must be made of protein. With its diverse and complex structure, protein seemed sophisticated enough to encode the vast information needed to direct life. But each time Avery's team destroyed proteins in their extracts, transformation continued unabated. Finally, they treated their extracts with an enzyme that specifically destroyed deoxyribonucleic acid, the DNA molecule that had seemed too simple and uniform to carry complex information. When DNA was destroyed, transformation ceased. The harmless bacteria remained harmless. The transforming principle, as Avery called it, was DNA. In 1944, Avery, MacLeod, and McCarty published their findings in the Journal of Experimental Medicine. Their paper was characteristically careful and cautious, presenting extensive evidence that DNA was the substance responsible for bacterial transformation. The chemical analyses were thorough, the experiments repeatable, the logic sound. Here was evidence that DNA, not protein, carried genetic information. Yet the scientific community hesitated. Many researchers remained skeptical. that DNA could be complex enough to encode heredity's intricate instructions. DNA contained only 4 types of building blocks, the nucleotide bases, while proteins contained 20 types of amino acids. How could a 4-letter alphabet carry as much information as a 20-letter one? Some scientists suggested that perhaps tiny amounts of protein contamination in Avery's extracts were actually responsible for transformation. Others simply found it difficult to abandon the protein theory after investing so much thought in that direction. Avery himself, ever cautious, never claimed with absolute certainty that DNA was the genetic material. He knew that extraordinary claims required extraordinary evidence, and he knew how easily scientists could be led astray by assumptions. But in private correspondence with his brother Roy, Avery allowed himself a moment of wonder. If DNA was truly the substance of genes, he wrote, it was possible to induce predictable and hereditary changes in cells, something tantamount to manipulating the very blueprint of life. The transformation experiments had opened a door, though not everyone was ready to walk through it. Evidence was accumulating, piece by careful piece, that this molecule called deoxyribonucleic acid held secrets worth pursuing. Other experiments would soon add weight to Avery's findings, making it increasingly difficult to ignore DNA's role in heredity. But for now, in the mid-1940s, the question lingered in laboratories around the world. If DNA truly was the molecule of heredity, what did it look like? How was it structured? And how could such a structure encode the infinite variation of living things? The stage was set for the next phase of discovery. Scientists would need to look more closely at DNA itself, to understand not just what it did, but how it was built. The chemical composition of this remarkable molecule would need to be examined with precision and care, revealing mathematical relationships that would point toward its hidden architecture. The evidence was mounting. By the late 1940s, experiments had pointed increasingly toward DNA as the carrier of genetic information, yet a profound mystery remained. Scientists knew that DNA existed in all living cells. They understood it was composed of 4 chemical building blocks called nucleotide bases: adenine, guanine, cytosine, and thymine. But how these components fit together, what pattern they formed, and why this particular molecule could store and transmit the intricate instructions for life, these questions hung in the air like morning mist, waiting for someone to bring clarity. Into this moment of uncertainty stepped a biochemist named Erwin Chargaff. Born in the Austro-Hungarian Empire in 1905, Chargaff had witnessed the great upheavals of the 20th century and carried with him a certain skepticism toward scientific orthodoxy. He had established himself at Columbia University in New York, where he studied the chemistry of nucleic acids with meticulous attention to detail. When he learned of Avery's work demonstrating that DNA carried genetic information, Chargaff experienced what he later described as a moment of revelation. If DNA truly was the molecule of heredity, then its chemical composition deserved far more careful scrutiny than it had previously received. The prevailing wisdom of the time held that DNA was a monotonous molecule, a simple repetitive structure that couldn't possibly encode complex information. Many scientists believed that the 4 bases occurred in equal proportions, arranged in a tedious, predictable pattern. This assumption had contributed to the widespread belief that proteins, with their 20 different amino acid building blocks, must be the true carriers of genetic information. But Chargaff suspected that the earlier measurements had been imprecise, conducted with techniques that weren't refined enough to detect subtle but meaningful differences. Beginning in 1947, Chargaff set about analyzing DNA samples from various organisms with unprecedented precision. He employed new methods of chemical separation called paper chromatography, combined with ultraviolet spectroscopy, to measure the exact quantities of each base present in DNA from different sources. His laboratory became a place of exacting measurement, where tiny samples were coaxed to reveal their secrets through patient, careful analysis. Each organism required multiple samples, multiple measurements, multiple confirmations. The work demanded a particular temperament, one that could find satisfaction in the accumulation of precise data points, building understanding one careful measurement at a time. What Chargaff discovered, as his data accumulated through months and years of work contradicted the accepted wisdom. The 4 bases did not occur in equal amounts. Different organisms had different proportions of the bases in their DNA. The DNA from human cells had a different composition than DNA from bacteria, which differed from DNA from yeast or plants. This variation itself was meaningful. It suggested that DNA might indeed be complex enough to encode different genetic information in different organisms. But within this diversity, Chargaff noticed something remarkable. A pattern emerged from his careful measurements, a mathematical relationship that held true across every organism he studied. In every sample from every source, the amount of adenine always approximately equaled the amount of thymine. Similarly, the amount of guanine always approximately equaled the amount of cytosine. The ratios weren't just close, they were strikingly precise, hovering around 1-to-1 with a consistency that couldn't be coincidental. These relationships, which became known as Chargaff's rules, represented a fundamental insight into DNA's composition. If adenine always matched thymine in quantity, and guanine always matched cytosine, this couldn't be mere chance. Some underlying structural principle must account for this mathematical regularity. The bases weren't arranged randomly or in simple repeating patterns. Instead, they appeared to be organized according to a specific pairing principle. Though what that principle might be, or what it revealed about DNA's structure, remained unclear. Chargaff published his findings in 1950, laying out the data with characteristic precision. The implications seemed profound, yet at first, relatively few scientists grasped their significance. The rules appeared to be an interesting chemical observation, a curious fact about DNA's composition, but their deeper meaning remained opaque. Chargaff himself understood that he had discovered something important, though he couldn't quite articulate what it meant for DNA's three-dimensional structure. He had identified a crucial piece of the puzzle, but the complete picture still eluded everyone. The patient work of measurement and analysis, conducted in a laboratory far from the spotlight, had revealed a hidden order in the molecule of heredity. DNA was not a monotonous, repetitive structure after all. It varied between organisms, yet followed strict internal rules. The 1-to-1 ratios suggested a kind of complementarity, a pairing principle that linked adenine with thymine and guanine with cytosine. Why this should be so, what structural arrangement could account for these numerical relationships, remained the question. As the data circulated through the scientific community, some researchers noted the findings with interest, while others overlooked their significance. The early 1950s found molecular biology at a threshold. Multiple pieces of evidence were accumulating: biochemical data like Chargaff's rules, physical measurements from other techniques, and theoretical considerations from various fields. Somewhere in all this information lay the answer to DNA's structure, waiting for someone to assemble the pieces correctly. Chargaff continued his work, measuring and analyzing, contributing to the growing body of knowledge about nucleic acids. His careful measurements had revealed a fundamental truth about DNA's composition, establishing ratios that any correct structural model would need to explain. The bases paired in specific ways, adenine with thymine and guanine with cytosine, in quantities that matched with mathematical precision. This complementarity pointed toward something essential about how DNA was organized, though the full significance of these pairings wouldn't become clear until the molecule's structure itself was finally revealed. The work of understanding DNA required contributions from many approaches: genetics, biochemistry, physics, and structural biology. Chargaff's meticulous chemical analysis had provided crucial constraints that the correct structure would need to satisfy. As other researchers pursued different methods of investigation, the rules he discovered would become one of several key insights that, when finally brought together, would unlock the secret of life's molecular blueprint. The molecule that carried the secret of heredity was now known to be DNA, thanks to the careful work of Avery and his colleagues, and its chemical composition had been precisely measured by Chargaff. Yet understanding what something is made of does not reveal its shape, and shape in the molecular world determines function. How were those four bases, adenine, thymine, guanine, and cytosine, arranged in space? What did a DNA molecule actually look like? To answer these questions, scientists would need to see the invisible, to perceive structures far smaller than any microscope could reveal. The technique that would unlock this mystery was X-ray crystallography, a method that sounds almost magical in its ability to reveal molecular architecture. When X-rays pass through a crystal, they scatter in precise patterns, determined by the arrangement of atoms within. These scattered rays create interference patterns that can be captured on photographic film, producing images of dark and light spots. To the trained eye, these patterns contain encrypted information about the three-dimensional structure of molecules. It was detective work of the highest order, requiring not just technical skill, but also intuition and patience. The physics behind this technique had been understood since the early 20th century, and by the 1950s, X-ray crystallography had already revealed the structures of various molecules and crystals. But DNA presented unique challenges. Creating suitable crystals or fibers of DNA required careful preparation, and interpreting the resulting X-ray patterns demanded both mathematical sophistication and physical insight. The photographs alone revealed nothing obvious. They were abstract patterns of spots and smudges that had to be decoded through calculation and reasoning. In London, at King's College, a young scientist named Rosalind Franklin had become intensely focused on applying X-ray crystallography to DNA. She had trained in physical chemistry in Cambridge and then spent several years in Paris, where she mastered the exacting techniques of X-ray diffraction. When she arrived at King's College in early 1951, she brought with her exceptional skill and a determination to achieve the highest quality results possible. Franklin approached scientific problems with precision and rigor, insisting on careful methodology and reproducible results before drawing conclusions. The DNA samples Franklin worked with came in two forms, which she designated A and B. The A form appeared when the DNA fibers were relatively dry, while the B form emerged when they absorbed moisture from the air. She recognized that these might represent different configurations of the same molecule, and she set about systematically studying both. Her laboratory notebooks reveal a methodical mind at work, carefully recording exposure times, humidity levels, and the quality of each photograph. She took hundreds of images, constantly refining her technique to capture clearer patterns. Working alongside Franklin at King's College was Maurice Wilkins, who had been studying DNA before her arrival. The working relationship between Franklin and Wilkins was complicated by unclear arrangements and misunderstandings about their respective roles and responsibilities. While Wilkins assumed they would collaborate, Franklin understood she had been given independent control over the X-ray work on DNA. This ambiguity created tension that would persist throughout Franklin's time at King's College, though both scientists continued their serious investigation of DNA's structure. Franklin's approach was to gather comprehensive data before proposing any structural model. She believed in letting the evidence accumulate, in allowing the X-ray patterns to speak for themselves through careful analysis. Throughout 1951 and into 1952, she produced increasingly refined images of both the A and B forms of DNA. Her photographs showed clear patterns, crosses and diamonds of spots, whose positions and intensities held clues about the underlying molecular arrangement. In May of 1952, Franklin captured an image that would become one of the most famous photographs in the history of science. Photograph 51, as it was labeled in her systematic numbering scheme, showed the B form of DNA with stunning clarity. The image displayed a distinctive X-shaped pattern of dark spots against a lighter background, with a clear pattern of reflections arranged in layers. To someone trained in interpreting such patterns, this photograph fairly shouted its message. The molecule was helical, a shape twisted like a spiral staircase or a coiled spring. The mathematics of helical diffraction were well established. When X-rays scatter from a helical structure, they produce exactly this kind of cross pattern. Franklin certainly recognized what photograph 51 revealed. Her notebooks from this period show calculations consistent with a helical structure, and she began working out precise measurements. The spacing of the spots indicated the pitch of the helix, how far the molecule advanced with each turn. The overall pattern provided information about the diameter of the structure. Layer by layer, the invisible was becoming visible through the language of X-ray diffraction. Franklin was also making progress understanding the A-form, which presented different challenges. By November 1952, she had given a seminar at King's College presenting her findings. She discussed the evidence for helical structure in the B-form and presented detailed measurements and calculations. Her approach remained cautious. She wanted to be absolutely certain of her conclusions before publishing, aware that premature claims could prove embarrassing if later evidence contradicted them. The X-ray images themselves were beautiful in their own way, abstract patterns that held concrete meaning for those who could read them. Each photograph represented hours of careful work, preparing the DNA fibers, adjusting the equipment, calculating the correct exposure time, developing the film in the darkroom. Franklin and her graduate student Raymond Gosling worked tirelessly to perfect their techniques, to coax ever clearer signals from their samples. The work demanded patience and precision, qualities Franklin possessed in abundance. What makes this period particularly significant in the history of science is that these X-ray photographs were providing crucial evidence about DNA's structure. Even as multiple teams raced toward a solution, the data Franklin was generating represented some of the best experimental evidence available about what DNA actually looked like at the molecular level. Photograph 51 was not merely a beautiful image, it was a Rosetta Stone for decoding the structure of the most important molecule in biology. In her systematic way, Franklin was building toward a comprehensive structural model based on solid evidence. She understood that DNA held the key to heredity, and she was determined to solve its structure correctly, with full scientific rigor. The X-ray patterns she and Gosling produced during 1951 and 1952 contained the essential clues needed to construct an accurate model of DNA. The detective work was yielding results, even as the race to solve the structure intensified at laboratories elsewhere, where other scientists were pursuing the same goal with different methods and increasing urgency. By the early months of 1951, the hunt for DNA's structure had become something more than academic curiosity. It had transformed into a race, though not everyone involved would have described it that way at the time. In laboratories scattered across 2 continents, scientists pursued the same prize, each bringing different tools and perspectives to bear on this fundamental question. The atmosphere crackled with possibility and urgency, as researchers sensed they stood on the threshold of a discovery that would reshape biology itself. In Pasadena, California, Linus Pauling worked with the confidence of someone who had already conquered similar challenges. He had won fame and accolades for determining the structure of proteins, discovering the alpha helix through a combination of model building and chemical intuition. Pauling possessed a rare gift for visualizing molecular architecture in 3 dimensions, seeing how atoms might arrange themselves in space to satisfy the requirements of chemistry. His approach relied less on experimental data and more on theoretical reasoning about what configurations would be chemically stable and elegant. He had proven this method spectacularly successful with proteins, and now he turned his formidable intellect toward DNA. Pauling's laboratory at the California Institute of Technology hummed with activity and innovation. He had access to excellent equipment, talented collaborators, and a reputation that opened doors throughout the scientific world. When Pauling focused his attention on a problem, other scientists took notice. His interest in DNA signaled that the time had come for this molecule to yield its secrets. Yet, despite his advantages, Pauling labored under one significant handicap. His political activities and outspoken views had attracted the attention of the United States government during an era of suspicion and scrutiny. When he planned to attend a scientific conference in England in 1952, the State Department denied his passport application, preventing him from traveling abroad. This restriction would keep him at a distance from crucial conversations and data emerging from British laboratories. Across the Atlantic at King's College London, Maurice Wilkins continued the X-ray crystallography work that had already produced tantalizing hints about DNA's structure. Wilkins had been among the first to obtain clear X-ray diffraction patterns from DNA fibers, recognizing their quality and potential. He possessed a methodical temperament, preferring careful, systematic progress over hasty conclusions. His laboratory had accumulated substantial data, and he understood that somewhere within those patterns lay the answer to DNA's architecture. The work demanded patience and precision, qualities Wilkins possessed in abundance. The situation at King's College had grown complicated, however, by questions of collaboration and credit. Rosalind Franklin had joined the laboratory and taken charge of the DNA X-ray work, bringing her exceptional technical skill and rigorous analytical approach. The relationship between Franklin and Wilkins had become strained, marked by misunderstandings about roles and responsibilities. Franklin worked independently, keeping her data close while she subjected it to thorough analysis. She distrusted premature speculation and resisted building models before the evidence warranted it. Her caution reflected scientific discipline, but it also meant that insights from her outstanding experimental work circulated less freely than they might have otherwise. Meanwhile, in Cambridge, two scientists had formed an unlikely partnership focused on the DNA problem. James Watson had arrived from America, a young biologist trained in genetics who had become fascinated by the challenge of determining molecular structures. Francis Crick was a physicist who had come to biology relatively late, bringing with him a gift for theoretical analysis and an infectious enthusiasm for scientific problems. Neither was supposed to be working on DNA. Their official projects lay elsewhere, yet they found themselves drawn together by shared curiosity and complementary skills. Watson and Crick made an odd pair in many ways. Watson, barely in his 20s, possessed an intense focus and competitive drive that sometimes struck others as brash. Crick, more than a decade older but still without his doctorate, was known for his booming laugh and willingness to challenge conventional thinking. What united them was a conviction that DNA's structure could be solved through model building, the approach that had served Pauling so well with proteins. Rather than waiting to accumulate complete experimental data, they believed in using available clues to construct physical models, testing whether proposed structures satisfied known chemical constraints and matched observed patterns. Their laboratory in Cambridge provided an environment conducive to this approach. The Cavendish Laboratory had a distinguished history in physics and a growing presence in molecular biology. Max Perutz and John Kendrew were already applying X-ray crystallography to protein structures, creating an atmosphere where structural questions dominated discussions. Watson and Crick inhabited this world as energetic participants, attending seminars, engaging in conversations over lunch, and gathering information from any available source. They read published papers carefully, discussed implications of various findings, and remained alert for any detail that might illuminate the DNA puzzle. The competitive dynamics among these groups remained somewhat hidden beneath the surface of collegial scientific discourse. Scientists shared information at conferences and through publications, yet they also guarded their most recent findings, hoping to be first to reach the goal. Pauling's interest raised the stakes considerably. His track record suggested he might solve DNA's structure through pure reasoning, much as he had divined protein architecture. Researchers at King's College possessed the best experimental data, but seemed to be moving cautiously. Watson and Crick had neither Pauling's reputation nor Franklin's data, but they had enthusiasm, theoretical insight, and a willingness to make bold guesses. As 1951 progressed into 1952, each group advanced along its chosen path. Pauling worked on his models in California, applying chemical principles to constrain possible structures. Franklin continued her meticulous X-ray studies in London, accumulating evidence and refining her understanding of DNA's characteristics. Wilkins pursued his own parallel investigations. Watson and Crick built models in Cambridge, incorporating insights gleaned from conversations, seminars, and published literature. The race had no official starting gun, no declared finish line, yet everyone involved sensed they were running toward something momentous. The scientific world watched with growing interest as the DNA problem attracted some of the brightest minds in molecular biology. Here was a mystery worthy of their talents, a fundamental question whose answer promised to illuminate the very basis of heredity and life itself. The molecule that carried genetic information from generation to generation surely held that information in some systematic way, encoded in its structure. Whoever first glimpsed that structure would gain insight into life's deepest mechanisms. The stage was set, the players assembled, and the critical information slowly accumulating, somewhere in the patterns of X-ray diffraction, in the ratios Chargaff had discovered, in the chemical properties of nucleotides and the physical constraints of molecular bonds, lay the answer. The race to find it had intensified, driven by competition, curiosity, and the profound human desire to understand the foundations of existence itself. The atmosphere in Cambridge during the early 1950s carried a particular energy, a sense that great discoveries were waiting just beyond the next careful observation, the next clever insight. In the Cavendish laboratory, a place already steeped in scientific history, two men had begun working together in an unusual partnership that would prove remarkably fruitful. James Watson had arrived in Cambridge as a young American, barely past his doctoral studies, with an intensity about him that some found abrasive, but which concealed a genuine passion for understanding life's fundamental mechanisms. Francis Crick, though still working toward his own PhD, despite being in his mid-30s, possessed a brilliant mind trained in physics and an infectious enthusiasm that expressed itself in loud, cheerful discussions that echoed through the laboratory corridors. Their supervisor, Max Perutz, had brought them together almost by accident, and neither man was officially supposed to be working on DNA at all, yet they found themselves drawn to the puzzle with increasing fascination. What made Watson and Crick's approach distinctive was their decision to build physical models, while others were conducting experiments, exposing DNA to X-rays, or performing chemical analyses. These two decided to work like architects, constructing three-dimensional representations using metal plates, wire, and colored balls mounted on rods. The technique had proven successful for Linus Pauling when he discovered the alpha helix structure of proteins, and Watson and Crick believed it might work for DNA as well. Their method required gathering information from many sources. They attended seminars where other scientists presented their findings. They read published papers carefully, extracting numerical data about bond angles and molecular distances. They engaged in long conversations over lunch, discussing what structures might be chemically possible and which seemed unlikely. Cambridge provided an ideal environment for this kind of work, where informal exchanges of ideas happened naturally in the corridors, at afternoon tea, or in the Eagle pub where the two often continued their discussions. Crucially, Watson and Crick were not conducting their own experimental work on DNA. They were synthesizers, bringing together pieces of information that others had discovered and trying to see how those pieces might fit together into a coherent whole. This approach had advantages and disadvantages. They could move quickly, testing ideas by building models rather than spending months on experiments. But they were also dependent on the accuracy and completeness of data from other sources, and they had to be careful not to overstep boundaries or claim credit for experimental work they hadn't done. The physical models they built served as three-dimensional hypotheses. Each model was a concrete proposal about how DNA's atoms might be arranged in space. Building the models forced them to confront practical constraints. Atoms could only bond at certain angles. Distances between molecules were fixed by chemical laws. If a model couldn't be built without distorting these fundamental rules, it couldn't be correct. The models turned abstract speculation into tangible objects that could be examined, critiqued, and improved. Their first serious attempt at building a DNA model came in late 1951, and it proved to be instructive in the way that mistakes often are. They constructed a structure with three intertwined chains and the phosphate groups positioned toward the center. Excited by their creation, they invited scientists from King's College London to come view it, including Maurice Wilkins and Rosalind Franklin. The meeting did not go well. Franklin, with her deep understanding of chemistry and her precise experimental data, quickly identified fatal flaws in their model. The water content was wrong, the phosphates were in an unlikely position, and the structure contradicted her X-ray diffraction data. Watson and Crick's supervisor, disappointed by this embarrassing episode, suggested they leave DNA work to the scientists who were actually conducting experiments on it. For more than a year, Watson and Crick dutifully turned their attention to other projects. Crick worked on his thesis about protein structure. Watson pursued studies related to tobacco mosaic virus. But neither man could completely let go of the DNA problem. They continued to think about it, to discuss possibilities, to watch for new information that might provide clues. What made their partnership effective was how their skills complemented each other. Watson brought his training in biology and genetics, understanding what any proposed DNA structure would need to accomplish biologically. He grasped that DNA had to carry information and must be capable of copying itself. Crick contributed his physicist's intuition about molecular structure and his ability to think through three-dimensional arrangements. He was particularly skilled at considering symmetry and at recognizing when proposed structures violated physical principles. They also brought different temperaments to their collaboration. Watson was more impulsive, quick to propose ideas and eager to move forward. Crick was more analytical, wanting to think through implications carefully. Watson pushed them forward, while Crick's critical thinking prevented them from pursuing too many dead ends. In their small shared office, they argued, agreed, disagreed again, sketched structures on paper, and gradually refined their understanding of what DNA's structure might look like. As 1952 turned into 1953, new information began flowing to them. Watson attended a research seminar and learned about Chargaff's rules regarding the equal proportions of certain bases. He had known about this work before, but hadn't fully appreciated its significance. Seeing the data presented again sparked new thoughts. Meanwhile, information about the X-ray photographs from King's College continued to circulate through scientific networks, including the crucial detail that DNA appeared to have a helical form. In Cambridge, with spring approaching and daffodils beginning to emerge in the college gardens, Watson and Crick sensed they were getting close to something important. They obtained permission to return to DNA model building, and the metal plates and wire molecules came out once more. The atmosphere in their office became charged with possibility. Each configuration they tried taught them something, ruling out certain arrangements, suggesting others worth exploring. The path to understanding DNA's structure was not a straight line, but rather a wandering journey with false starts, corrections, and gradually accumulating insights. Watson and Crick were bringing together threads from multiple sources, weaving them into a coherent picture. The physical models they built were not just representations of molecules, but thinking tools, ways to make the invisible visible and the abstract concrete. In that small Cambridge office, amid the clutter of metal and wire, one of biology's greatest puzzles was approaching its solution. The winter morning of February 28, 1953, began like many others in Cambridge, but by lunchtime, everything had changed. In their small office at the Cavendish Laboratory, James Watson and Francis Crick had finally assembled the pieces of a puzzle that had consumed them for months. The answer, when it came, arrived with such elegant simplicity that it seemed almost inevitable in retrospect. Watson had arrived at the lab that Saturday morning with fresh insight from a late-night session of shuffling cardboard cutouts representing the chemical bases of DNA. He had been pairing them in a new way, not like with like as he had previously attempted, but adenine with thymine and guanine with cytosine. The shapes fit together beautifully, and more remarkably, each pair was exactly the same width. This symmetry meant that a regular helical structure could accommodate these base pairs without distortion. When Crick arrived and saw Watson's arrangement, he immediately grasped its significance. This pairing scheme honored Chargaff's rules, which had shown that the amount of adenine in DNA always equals thymine, and guanine always equals cytosine. But it did more than that. It explained why those rules existed in the first place. The bases weren't just floating randomly. They were paired in complementary fashion, locked together by hydrogen bonds in the center of the double helix. Together they began constructing a physical model, using metal plates and rods to represent the atoms and bonds. The structure that emerged was breathtaking in its elegance. Two sugar-phosphate backbones spiraled on the outside, like the twisted railings of a spiral staircase. Between them, the paired bases formed the steps, holding the two strands together. The entire molecule twisted in a gentle right-handed helix, making one complete turn every 10 base pairs. The beauty of the structure lay not just in its form, but in what it immediately suggested about function. If adenine always paired with thymine, and guanine always paired with cytosine, then each strand of the double helix contained the complete information needed to create the other strand. The two strands were complementary, like a photograph and its negative. This meant that DNA could copy itself simply by separating the two strands and using each as a template to build a new partner strand. Watson and Crick could hardly contain their excitement as they examined their model from every angle, checking that it satisfied all the known chemical and physical constraints. The model explained Rosalind Franklin's X-ray data, which had shown the helical structure with its regular repeating pattern. It honored the chemical rules about how bases could bond with each other. It made sense of Chargaff's mysterious one-to-one ratios. Every piece of evidence fit. That lunchtime, Crick's voice rang out in the Eagle pub near the laboratory, announcing to anyone within earshot that they had discovered the secret of life. It was an audacious claim, perhaps premature, yet not entirely wrong. The structure they had found would indeed prove to be the foundation for understanding how living things store, copy, and pass on genetic information. In the days that followed, they refined their model and began writing a paper to announce their discovery. They invited colleagues to examine the structure, including Maurice Wilkins, who had been working on DNA at King's College London. The model withstood scrutiny. Its simple elegance made it convincing. Unlike earlier attempts to solve DNA's structure, this one felt right to those who understood the chemistry and physics involved. The paper they submitted to the journal Nature was remarkably brief, barely more than a page, published on April 25, 1953. It described the double helical structure with characteristic British understatement. Near the end, Watson and Crick included a sentence that has become famous for its restrained yet pregnant phrasing. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. In those carefully chosen words, they hinted at the profound implications of their discovery, without overstating their case. The same issue of Nature included papers by Wilkins and his colleagues, and by Rosalind Franklin and her student Raymond Gosling, presenting the X-ray crystallography data that supported the double helix model. These experimental results provided the evidence that confirmed Watson and Crick's theoretical structure. The convergence of model building and experimental data created a complete and compelling picture. Not everyone immediately recognized the significance of the discovery. Science often advances in ways that seem obvious only in hindsight, but gradually, the community of biologists and chemists came to appreciate that something fundamental had been revealed. The double helix provided a molecular explanation for how inheritance works, how mutations occur, and how evolution proceeds through changes in DNA sequences. The structure answered old questions while raising new ones. If the sequence of bases encoded genetic information, how exactly did those sequences specify the characteristics of living things? How did cells read the information in DNA and use it to build proteins? The double helix opened doors to these deeper questions, providing the foundation for the molecular biology revolution that would unfold in the decades ahead. That February morning in Cambridge marked a turning point in humanity's understanding of life itself. Two scientists, working with information gathered by many others, had revealed the architecture of heredity. The twisted ladder of the double helix, with its complementary base pairs forming the rungs, would become one of the most recognizable symbols in science. In its elegant simplicity lay the explanation for the continuity of life across generations and the diversity of life across species. The code was written in 4 chemical letters, arranged along the rails of a spiral staircase, carrying the instructions that make each living thing what it is. The elegant structure that Watson and Crick had assembled from wire and metal plates held within it a secret that was almost immediately apparent to anyone who understood what they were looking at. The double helix was beautiful, yes, but it was also profoundly practical. The arrangement of the 2 strands, wound around each other with their complementary bases paired in the middle, suggested something remarkable. It suggested how life could copy itself. Consider the challenge that every living cell faces. Before a cell divides, it must duplicate its genetic information so that each daughter cell receives a complete set of instructions. For generations, Scientists had wondered how this copying process could occur with such fidelity that traits passed accurately from parent to offspring, from cell to cell, across countless generations. The answer lay in those paired bases running up the center of the double helix like rungs on a twisted ladder. Watson and Crick recognized this immediately. In their famous paper published in Nature in April 1953, they included a sentence of remarkable understatement. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. This modest phrase hinted at something extraordinary. The structure itself revealed how DNA could replicate. The mechanism relied on the complementary nature of the base pairs, adenine always paired with thymine, and guanine always paired with cytosine. This wasn't arbitrary. The shapes and chemical properties of these molecules meant they fit together in these specific combinations, forming stable hydrogen bonds that held the two strands together. If you knew the sequence of bases on one strand, you automatically knew the sequence on the other strand. They were complements, mirror images in a sense, each containing the information needed to recreate the other. Imagine the double helix beginning to unwind, like a zipper being pulled apart. As the two strands separate, each becomes a template. Where there was an adenine on the original strand, a new thymine could attach. Where there was a guanine, a new cytosine could find its place. Each old strand could direct the assembly of a new partner strand, following the simple rules of base pairing. When the process finished, there would be two double helices, where before there had been one, each containing one strand from the original molecule and one newly synthesized strand. This semi-conservative model of replication, as it came to be called, was elegant in its simplicity. Each new DNA molecule conserved half of the original, passing one strand to each daughter molecule. The process ensured both continuity and accuracy. The genetic message could be preserved across generations because the template mechanism built error correction into the very structure of the molecule. But recognizing that this mechanism was possible was different from proving that it actually occurred this way in living cells. Through the late 1950s, scientists worked to demonstrate that DNA replication actually proceeded as Watson and Crick had suggested. The decisive experiment came from Matthew Meselson and Franklin Stahl at the California Institute of Technology in 1958. Their experimental approach was ingenious. They grew bacteria in a medium containing a heavy isotope of nitrogen, nitrogen-15, which the bacteria incorporated into their DNA. This heavy DNA could be distinguished from normal DNA containing the common nitrogen-14 isotope because it had a different density. After allowing the bacteria to grow for several generations with the heavy nitrogen, Meselson and Stahl transferred them to a medium containing only normal nitrogen-14. The question was what would happen when these bacteria with heavy DNA replicated their genetic material in the presence of only light nitrogen. If replication was semi-conservative, as Watson and Crick predicted, the first generation of new DNA molecules should be hybrid, each containing one heavy strand from the original DNA and one light strand newly synthesized from the light nitrogen. If DNA copied itself by some other mechanism, the results would be different. When Meselson and Stahl examined the DNA after one round of replication, they found exactly what the semi-conservative model predicted. The DNA had an intermediate density, consistent with molecules containing one heavy and one light strand. After a second round of replication, they found a mixture, some DNA with intermediate density and some with fully light density. The pattern matched the predictions perfectly. DNA replicated by unwinding and using each strand as a template, just as the double helix structure suggested. The machinery that accomplishes this replication in living cells is intricate and remarkable. Enzymes called helicases move along the double helix, breaking the hydrogen bonds between base pairs and unwinding the strands. Other enzymes called DNA polymerases move along each separated strand, reading the sequence and adding complementary nucleotides to build the new strand. The process occurs with astonishing speed and accuracy, with multiple replication points proceeding simultaneously along large chromosomes. This replication mechanism explained something fundamental about heredity. When cells divide, they pass identical genetic information to their descendants, because DNA can serve as its own template. When organisms reproduce, they pass genetic information to their offspring through DNA molecules that have been copied using this same template mechanism. The continuity of life itself depends on the ability of DNA to direct the synthesis of accurate copies of itself. The implications reached even further. The template mechanism meant that changes in DNA sequence, mutations, would also be faithfully copied. If a mistake occurred during replication, or if DNA was damaged by radiation or chemicals, that altered sequence could be passed on to future generations. This provided a molecular explanation for genetic variation, the raw material on which natural selection acts. The mechanism of replication thus connected to the mechanism of evolution. As scientists in laboratories around the world absorbed the meaning of the double helix structure, they began to ask the next logical questions. DNA could copy itself, but how did it actually do anything? How did this molecule, composed of just 4 different bases arranged in varying sequences, direct the building of organisms? How did the information encoded in DNA's 4-letter alphabet translate into the proteins that make up living things? The structure of DNA had been revealed, but its function remained mysterious. The answer to those questions would require another decade of intensive research, drawing together insights from biochemistry, genetics, and molecular biology. But now at least scientists knew they were asking the right questions. They had found the molecule of heredity, understood its structure, and discovered how it copies itself. The next challenge was to understand how DNA's information becomes life. The double helix had revealed its elegant structure, showing how genetic information could be copied and passed from one generation to the next. Yet knowing the shape of DNA was only part of the story. A profound mystery remained, one that would occupy some of the finest minds in molecular biology throughout the late 1950s and into the 1960s. The question was both simple and extraordinarily complex. How does a molecule with just 4 different letters spell out instructions for building proteins made from 20 different amino acids? Imagine trying to write the complete works of Shakespeare using only 4 letters of the alphabet. This was essentially the challenge that DNA faced, and that scientists needed to understand. The 4 bases, adenine, thymine, guanine, and cytosine, somehow had to encode instructions for assembling proteins from a palette of 20 distinct amino acids. The mathematics seemed puzzling at first. If each base coded for 1 amino acid, you could only specify 4 different amino acids. If pairs of bases formed the code, you could specify 16 combinations. Still not enough, but 3 bases in a row could create 64 different combinations, more than enough to handle 20 amino acids. This theoretical possibility of a triplet code was recognized fairly early, but proving it and actually breaking the code required years of ingenious experimentation. The work took place in laboratories across the world, with scientists developing new techniques to tease out how nature had written its instruction manual. Francis Crick, who had helped discover the double helix structure, became deeply interested in the coding problem. He brought his characteristic clarity of thought to the challenge, proposing ideas about how the code might work. Working with other scientists, he helped establish that the genetic code was indeed read in groups of 3 bases, which came to be called codons. Each codon would specify 1 amino acid in a protein chain. But which codon specified which amino acids? This required actually reading the code, and that breakthrough came from an American biochemist named Marshall Nierenberg, working at the National Institutes of Health in Bethesda, Maryland. In 1961, Nierenberg and his colleague Heinrich Mathieu performed a remarkably clever experiment. They created an artificial strand of RNA containing only 1 type of base, uracil, which in RNA takes the place of thymine. This artificial message, called poly-U, was then added to a cell-free system containing all the molecular machinery needed to make proteins. The result was striking. The poly-U message directed the creation of a protein chain made entirely of 1 amino acid, phenylalanine. The first word in the genetic dictionary had been read. The codon UUU meant phenylalanine. It was a moment of profound discovery. though one that happened quietly in a laboratory rather than with fanfare. Nirenberg announced his findings at an international conference in Moscow, and suddenly the race was on to decipher the rest of the code. Other scientists joined the effort with enthusiasm. Nirenberg continued his work, creating artificial RNA messages with different combinations of bases. Gobind Khorana, working at the University of Wisconsin, developed techniques to synthesize RNA molecules with specific repeating sequences. By carefully analyzing what proteins these artificial messages produced, he could deduce which codons specified which amino acids. It was painstaking work, requiring careful chemistry and patient analysis, but piece by piece, the codebook was being filled in. Severo Ochoa at New York University also contributed to cracking the code, using enzymes to create RNA molecules that could be tested in protein synthesis systems. The competition was friendly but intense, with laboratories sharing findings at scientific meetings and through publications. The genetic code was becoming legible, word by word. By the middle of the 1960s, the entire genetic code had been worked out. All 64 possible three-letter codons had been assigned their meanings. The code proved to have elegant features. Most amino acids were specified by more than one codon, a redundancy that provided some protection against errors. 3 codons didn't specify any amino acid at all. Instead, they served as punctuation marks, stop signals that told the cellular machinery when a protein chain was complete. One codon, AOG, had a dual function, coding for the amino acid methionine, but also serving as a start signal, marking where protein synthesis should begin. Perhaps the most remarkable discovery about the genetic code was its universality. The same codons specified the same amino acids in bacteria, in plants, in fungi, in animals. A codon that meant phenylalanine in a bacterium also meant phenylalanine in a human cell. This universal language suggested that all life on Earth descended from a common ancestor that had established this coding system billions of years ago. The code had been so successful, so fundamental to life, that it had been conserved across the vast spans of evolutionary time. The cracking of the genetic code represented a triumph of molecular biology, but it was also a beginning. Knowing the codebook meant that scientists could now read DNA sequences and predict what proteins they would produce. They could understand how mutations, changes in DNA letters, might alter proteins and cause disease. They could begin to manipulate the code, inserting genes from one organism into another and having them properly read and expressed. Nirenberg received the Nobel Prize in Physiology or Medicine in 1968, sharing it with Khorana and Robert Holley, who had worked out the structure of transfer RNA, another crucial piece of the protein synthesis machinery. The recognition came swiftly, because the importance of the work was immediately apparent. The genetic code was fundamental to understanding how life worked at its most basic level. As scientists absorbed this new knowledge, they marveled at the elegance of the system. 4 simple bases, arranged in groups of 3, could specify all the diversity of proteins that make life possible. The same code operated in every cell of every organism, a universal language of life written in chemical letters. It was as if nature had invented a digital information system billions of years before humans developed computers and binary code. The story of how cells actually use this code, how DNA is transcribed into RNA, and how RNA is translated into proteins, would continue to unfold in the years that followed. But the basic cipher had been broken. The 4-letter alphabet of DNA and its translation into the 20-letter alphabet of proteins was no longer a mystery. Life's instruction manual could finally be read. With the genetic code now deciphered, scientists understood how 3-letter words spelled in DNA's 4-letter alphabet could specify the 20 amino acids that build proteins. But knowing the dictionary was only part of the story. The question remained, how does the cell actually use this information? How does the code written in DNA, safely stored in the nucleus, direct the construction of proteins out in the cellular workspace? The answer would reveal an elegant molecular dance, a flow of information that occurs in every living cell countless times each day. The first insight came from recognizing that DNA itself doesn't leave the nucleus. Like a precious original manuscript kept in a library's restricted collection, the DNA remains protected, never venturing out into the cell's bustling cytoplasm, where proteins are actually assembled. Instead, the cell creates copies of the instructions it needs, temporary working documents, that can travel safely, while the original stays secure. This is where RNA enters the story. Ribonucleic acid, DNA's close chemical cousin, differs in just a few key ways. Where DNA uses the sugar deoxyribose, RNA uses ribose. Where DNA's double helix remains tightly wound, RNA typically exists as a single strand. And where DNA uses the base thymine, RNA substitutes a similar base called uracil. These differences make RNA perfect for its role as a molecular messenger. The process begins with transcription, a word borrowed from the scribes who copy manuscripts by hand. Inside the nucleus, when a particular protein needs to be made, the section of DNA containing that gene begins to unwind. The two strands of the double helix separate, exposing the bases along one strand. An enzyme called RNA polymerase moves along this exposed strand, reading the sequence of bases and constructing a complementary strand of RNA. As the RNA polymerase travels along the DNA template, it builds the RNA copy one letter at a time. Where it encounters an adenine in the DNA, it adds a uracil to the growing RNA strand. Where it finds a thymine, it adds an adenine. Guanine pairs with cytosine, and cytosine with guanine, just as in DNA replication. The result is a single-stranded RNA molecule that carries the same information as the gene, a working copy of the instructions. This messenger RNA, or mRNA as scientists call it, detaches from the DNA once transcription is complete. The DNA strands rejoin, reforming the double helix, while the newly created mRNA molecule makes its way out of the nucleus. It passes through pores in the nuclear membrane and enters the cytoplasm, carrying its encoded message to the sites where proteins are built. These protein factories are structures called ribosomes, complex molecular machines made of both RNA and protein. Each ribosome is like a tiny workshop, equipped to read the mRNA instructions and assemble amino acids in the precise order specified by the genetic code. When an mRNA molecule reaches a ribosome, the ribosome binds to it and begins reading the sequence of bases, 3 letters at a time. But the ribosome cannot simply translate codons into amino acids on its own. It needs another type of RNA to bring the amino acids to the assembly line. Transfer RNA, or tRNA, serves this crucial role. Each tRNA molecule is folded into a characteristic shape, with one end that recognizes a specific 3-letter codon, and another end that carries the corresponding amino acid. There are different tRNA molecules for each amino acid, each one matching the codon that specifies its particular amino acid. As the ribosome moves along the mRNA, reading codon after codon, the appropriate tRNA molecules arrive in sequence. Each tRNA brings its amino acid and pairs its recognition sequence with the matching codon on the mRNA. The ribosome then catalyzes the formation of a chemical bond between adjacent amino acids, linking them together like beads on a string. Once a tRNA has delivered its amino acid, and that amino acid has been added to the growing chain, the tRNA releases and floats away, ready to pick up another amino acid and repeat its delivery service. This process of translation continues, codon by codon, amino acid by amino acid, until the ribosome encounters one of the stop codons. These special 3-letter sequences signal that the protein is complete. The ribosome releases the finished chain of amino acids, and that chain folds into its specific 3-dimensional shape, becoming a functional protein, ready to carry out its role in the cell. The entire pathway from DNA to RNA to protein came to be known as the central dogma of molecular biology, a term coined by Francis Crick in the late 1950s. Information flows in one direction, from DNA to RNA to protein. DNA preserves the information across generations. RNA carries working copies of specific instructions. Proteins do the actual work of the cell, serving as enzymes, structural components, signaling molecules, and countless other functions. This understanding revealed the profound unity of life at the molecular level. Every organism, from bacteria to humans, uses the same basic mechanism. The genetic code is nearly universal, meaning the same codons specify the same amino acids in virtually all living things. An mRNA sequence from a human cell, if placed in a bacterial cell, would be read the same way and produce the same protein. This universality suggested that all life on Earth descended from a common ancestor that used this system billions of years ago. The discovery of how information flows from DNA to proteins also explained how cells can be so different from one another, despite having identical DNA. A liver cell and a brain cell in the same person contain exactly the same genetic information, but they look and function completely differently because they transcribe different sets of genes. Each cell type activates only the genes relevant to its function, creating the proteins it needs, while leaving other genes silent. As scientists observed these processes more closely, they discovered layers of complexity and regulation. The original mRNA transcript in eukaryotic cells often undergoes editing, with some sequences removed and others retained before the final message travels to the ribosomes. Regulatory proteins control which genes get transcribed, and when small RNA molecules can interfere with translation, fine-tuning which proteins get made. The simple flow from DNA to RNA to protein branched into an intricate network of control and modification. Yet beneath all this complexity, the fundamental pathway remains elegant and universal. 4 letters in DNA become 4 letters in RNA become 20 amino acids in proteins. From this chemical simplicity emerges all the complexity of life, all the diversity of form and function that fills our world. The discovery of this pathway completed a crucial chapter in understanding life's molecular foundation, revealing how the information encoded in DNA's double helix actually manifests as the living, breathing, functioning organisms we observe all around us. The understanding of how DNA encodes life and directs the creation of proteins had opened extraordinary doors, but a grander question beckoned from the horizon. If scientists could read the genetic code in principle, could they read the entire instruction manual of a human being? Could they spell out, letter by letter, all 3 billion base pairs that make up human DNA? The very idea seemed almost absurdly ambitious when it was first seriously proposed in the mid-1980s. The notion of reading the complete human genome emerged gradually from conversations at scientific meetings and conferences. Researchers had already sequenced small stretches of DNA from various organisms, including humans. They had decoded individual genes, learning the precise order of letters that spelled out instructions for specific proteins. But the human genome contained an estimated 3 billion letters. The task of reading them all seemed to belong more to the realm of science fiction than practical science. Yet the idea persisted, growing stronger with each passing year. In 1990, the Human Genome Project officially launched as an international scientific collaboration with an audacious goal to determine the complete sequence of all the DNA in human chromosomes within 15 years. The project would require unprecedented coordination between research centers across multiple countries, the development of new technologies, and funding on a scale rarely seen in biological research. The technical challenges were formidable. The methods available in 1990 for reading DNA sequences were painstakingly slow. A single researcher might sequence a few hundred base pairs in a day of careful laboratory work. At that rate, reading 3 billion letters would take centuries. The project would succeed or fail based on whether new technologies could be developed to dramatically accelerate the process. The approach chosen was both practical and methodical. Rather than trying to read the genome from beginning to end like a book, scientists would break chromosomes into thousands of smaller, overlapping fragments. Each fragment would be sequenced separately. Then sophisticated computer programs would identify the overlaps and piece everything back together, like assembling an enormous jigsaw puzzle with 3 billion pieces. Research centers in the United States, United Kingdom, France, Germany, Japan, and China took on different portions of the genome. Each center developed its own expertise and refined techniques for preparing DNA, running sequencing reactions, and analyzing results. The collaborative spirit of the project meant that data was shared openly, with sequences deposited into public databases where scientists worldwide could access them. As the 1990s progressed, the technology improved steadily. automated sequencing machines replaced much of the manual laboratory work. These machines could process multiple samples simultaneously, reading the DNA sequences and storing the results directly in computer files. The pace of sequencing accelerated from thousands of base pairs per day to millions per day as the technology matured. In 1995, the project achieved an important milestone by completing the first full genome sequence of a free-living organism, a bacterium called Haemophilus influenzae. Though much smaller than the human genome at about 1.8 million base pairs, this achievement demonstrated that complete genome sequencing was possible and provided valuable lessons about how to organize and analyze such vast amounts of genetic data. The late 1990s brought unexpected competition to the publicly funded Human Genome Project. A scientist named Craig Venter founded a private company called Celera Genomics with the aim of sequencing the human genome using a different, faster approach. Rather than carefully mapping where each fragment belonged before sequencing it, Venter's team would sequence millions of fragments and use powerful computers to assemble them, based on overlaps alone. This shotgun approach was riskier, but potentially much faster. The race between the public project and private company added drama and urgency to the work. Some worried that if a private company finished first, they might patent the genome sequence or restrict access to the data. Others welcomed the competition as spurring both efforts to move faster. The pace of sequencing accelerated dramatically as both groups pushed their technologies to the limit. By the spring of 2000, both groups had assembled working drafts covering most of the human genome. In June of that year, leaders of the public and private efforts stood together at the White House for a carefully orchestrated announcement. The first draft of the human genome had been completed, a moment of scientific achievement that transcended the competition between the groups. The draft announced in 2000 was incomplete, with gaps remaining in difficult-to-sequence regions, and some sections still being refined. The work continued, filling in gaps and improving accuracy. In 2003, timed to coincide with the 50th anniversary of Watson and Crick's double helix structure, the Human Genome Project was declared complete. The final sequence covered about 99% of the gene-containing portions of the genome, with an accuracy of 99.99%. Reading the human genome had required the efforts of thousands of scientists, technicians, and computer specialists working across more than a decade. The cost exceeded $3 billion, roughly $1 per base pair. But the achievement represented more than just an impressive feat of coordination and technological development. The genome sequence was a fundamental reference work, a complete genetic blueprint that would enable countless future discoveries. With the human genome now readable, scientists could search for genes associated with diseases, compare human DNA to that of other species, and understand how genetic variations among individuals influence traits and health. The raw sequence was like a vast encyclopedia written in a language scientists were still learning to fully comprehend, but having the complete text available opened possibilities that previous generations could barely have imagined. The technology that had been developed to sequence the human genome continued to improve after the project's completion. Methods became faster and cheaper at an astonishing rate. What had cost billions of dollars and taken years in 2000 could be accomplished for thousands of dollars in a matter of days by 2010, and for hundreds of dollars in hours by 2020. As night settles softly around us, we can reflect on this remarkable achievement. The human genome, read letter by letter, stands as a monument to scientific collaboration and determination. 3 billion base pairs, each now known and recorded, waiting to reveal their secrets to the next generation of curious minds seeking to understand the blueprint of human life. The completion of the first draft of the human genome in the year 2000 was like finally opening a book that had been locked away for millions of years. Scientists around the world gathered around their computer screens, eager to read what was written in those 3 billion letters of genetic code. They expected to find certain things, predictions made from decades of biological research. But the genome, as it turned out, was full of surprises. One of the first unexpected discoveries was a matter of simple counting. For years, scientists had estimated that humans must have somewhere between 80,000 and 140,000 genes. After all, we are complex creatures with intricate brains, sophisticated immune systems, and elaborate developmental programs. It seemed reasonable that such complexity would require a vast number of genetic instructions, but as the genome sequencing teams tallied up the actual number of protein-coding genes, they arrived at a figure far lower than anyone had anticipated. Humans have only about 20,000 to 25,000 genes, not so very different from the humble roundworm with its 20,000, or the fruit fly with its 14,000. This revelation was humbling in a way. Complexity, it seemed, did not arise simply from having more genes. Instead, the genome suggested that the same genes could be used in different combinations, at different times, in different amounts, to create the remarkable diversity of human biology. A single gene might produce multiple versions of a protein through a process called alternative splicing, where different segments of the genetic message are included or excluded. The genome was not just a simple list of instructions, but a flexible script that could be read in many ways. Another surprise awaited in the vast stretches of DNA between the genes themselves. Only about 2% of the human genome consists of sequences that directly code for proteins. 2%. The remaining 98% was initially dismissed by some as junk DNA, evolutionary debris accumulated over millions of years. But as scientists looked more carefully, they began to find that much of this non-coding DNA was anything but junk. Hidden within these sequences were regulatory elements, switches and dimmers, that controlled when and where genes were turned on or off. Some of these regulatory regions were located far from the genes they controlled, sometimes millions of letters away along the chromosome. The genome revealed itself to be not just a collection of genes, but an elaborate control system for managing those genes. Scattered throughout the genome, researchers found the remnants of ancient viral infections, retroviruses, which insert their genetic material into their host's DNA, had left their marks throughout human chromosomes. These viral sequences made up about 8% of the human genome. Evolutionary fossils from infections that occurred millions of years ago in our distant ancestors. Most of these viral remnants were now inactive, their ability to replicate long since lost to mutation. But they remained as a record of ancient biological struggles, battles fought at the molecular level, long before humans walked the earth. The genome also revealed how remarkably similar we are to other species. Humans share about 99% of their DNA sequence with chimpanzees, our closest living relatives. The genetic difference between us and our primate cousins amounted to only about 1%, a slim margin that somehow encompasses all the distinctions we consider uniquely human. Looking further back in evolutionary time, humans share about 90% of their genes with mice, and about 60% with fruit flies. Even baker's yeast, a single-celled fungus, shares about 40% of its genes with humans. These comparisons told a story of common ancestry, of genetic toolkits conserved and modified over hundreds of millions of years of evolution. Within the human population itself, the genome revealed how similar we all are to one another. Any two humans, chosen at random from anywhere on Earth, are about 99.9% identical in their DNA sequences. The variation that accounts for all our individual differences, all the diversity of human appearance and traits, resides in just 0.1% of our genetic code. This tiny fraction of difference reminded scientists that the categories we create to divide humanity are far less significant than the overwhelming genetic unity that binds us together. The genome also contained regions of surprising repetition, tandem repeats, where the same short sequence appears over and over again, were scattered throughout chromosomes. Some genes themselves appeared in multiple copies, sometimes in clusters on the same chromosome, sometimes scattered across different chromosomes. These duplications told stories of evolutionary history, of genes that had been copied and then modified to take on new or slightly different functions. Gene duplication, the genome suggested, was one of evolution's strategies for innovation. As researchers compared genomes from different individuals, they mapped regions where genetic variation was particularly common. These single nucleotide polymorphisms, places where different people might have different letters in their genetic code, occurred about once every 1,000 letters or so. Some of these variations had no discernible effect, falling in regions that didn't code for anything critical. Others subtly altered protein function, or changed how much of a protein was made. These variations contributed to individual differences in traits, ranging from height to susceptibility to certain diseases. The genome revealed stretches where genes were densely packed together, regions rich in genetic information, and other stretches that seemed relatively barren, with long expanses containing few genes. The distribution was uneven, patchy, like settlements scattered across a landscape. Some regions were hotspots for recombination, where chromosomes frequently exchanged segments during the formation of egg and sperm cells. Other regions rarely recombined, remaining stable across generations. Reading the genome of humans naturally led to reading the genomes of other species. Each genome sequenced added to a growing library of genetic information, allowing comparisons that illuminated evolutionary relationships and revealed which genes were essential across all life and which were unique to particular lineages. The genomes of plants showed different organizations and strategies from animals. Bacterial genomes, compact and efficient, stood in contrast to the sprawling genomes of many animals with their abundance of non-coding sequence. What the genome told us, ultimately, was that heredity was more intricate than anyone had imagined. The simple 4-letter code contained layers of meaning and regulation, overlapping messages and ancient history. It was a text that had been written, edited, and revised over billions of years, carrying within it the story of life's journey from single cells to the magnificent diversity of the living world. Understanding the sequence was just the beginning. Learning to read its deeper meanings, to understand how context and timing turned those letters into living organisms, would occupy scientists for generations to come. The knowledge gained from reading the human genome opened doors that had previously existed only in imagination. Understanding the sequence of 3 billion letters was one achievement, but learning to use that knowledge to heal, to diagnose and to produce medicines, transformed molecular biology from a purely academic pursuit into something with immediate practical applications. The story of DNA moved from laboratories focused on basic research into hospitals, pharmacies, and medical clinics around the world. One of the earliest medical applications emerged from simply reading specific sections of a person's DNA. Genetic testing allowed physicians to examine particular genes known to be associated with certain conditions. Some genetic disorders result from mutations in single genes, and identifying these mutations became possible once scientists knew what sequences to look for. Tests for conditions like cystic fibrosis, sickle cell anemia, and Huntington's disease could now be performed by examining DNA samples, providing information that was previously unobtainable until symptoms appeared, if they appeared at all. This ability to read genetic variations opened new approaches to understanding disease risk. Some mutations significantly increase the likelihood of developing certain conditions later in life. The BRCA1 and BRCA2 genes, when carrying particular mutations, substantially elevate breast and ovarian cancer risk. Discovering these mutations allows individuals to make informed decisions about monitoring and prevention strategies. The information contained in DNA sequences began informing healthcare decisions in ways that would have seemed like science fiction just decades earlier. Beyond diagnosis, DNA technology revolutionized the production of medicines themselves. Before molecular biology techniques matured, certain essential medicines were difficult or impossible to produce in sufficient quantities. Insulin, the hormone crucial for managing diabetes, was originally extracted from the pancreases of pigs and cattle. This process was expensive, produced limited quantities, and sometimes caused immune reactions because animal insulin differs slightly from human insulin. The solution came from enlisting bacteria as molecular factories. Scientists learned to insert the human insulin gene into bacterial DNA, transforming these microorganisms into producers of genuine human insulin. The bacteria, following their genetic instructions as they always do, read the inserted human gene and manufactured human insulin protein. These microscopic workers could be grown in large fermentation tanks, producing abundant quantities of identical, pure human insulin. The first genetically engineered human insulin reached patients in 1982, marking a milestone in biotechnology's practical applications. This approach extended far beyond insulin. Growth hormone, previously extracted in tiny amounts from human cadavers, could now be produced by engineered bacteria. Clotting factors for hemophilia patients, interferon for treating certain diseases, and numerous other proteins became available through similar techniques. DNA technology had created an entirely new pharmaceutical industry, based on reading genetic sequences and using that information to manufacture complex biological molecules. As scientists learned more about how genetic variations affect drug responses, the concept of personalized medicine began to emerge. People metabolize medications at different rates, depending on variations in their genes. Some individuals process certain drugs quickly, requiring higher doses, while others metabolize the same drugs slowly, risking toxic buildup at standard doses. Genetic testing could identify these variations, allowing physicians to tailor prescriptions to individual genetic profiles, rather than relying on one-size-fits-all dosing. Cancer treatment particularly benefited from understanding DNA at the molecular level. Tumors arise from accumulated mutations in DNA, and different tumors carry different combinations of mutations. By sequencing tumor DNA, oncologists could identify which specific mutations were driving a particular patient's cancer. This information guided treatment selection, choosing therapies most likely to be effective against that specific combination of genetic changes. The 4 letters of DNA were being read to create treatment strategies customized to individual patients. Pharmacogenomics emerged as a field dedicated to understanding how genetic variation affects drug response. The enzyme that metabolizes clopidogrel, a medication preventing blood clots, works differently in people carrying certain genetic variants. Some patients gain little benefit from standard doses because their bodies cannot activate the drug efficiently. Genetic testing identifies these individuals, allowing physicians to select alternative medications or adjust dosing. Similar principles apply to numerous medications across many disease categories. DNA technology also transformed the search for new medicines. Understanding the precise molecular mechanisms of diseases revealed through genetic research identified new targets for drug development. Rather than testing thousands of compounds hoping to find something effective, researchers could design molecules intended to interact with specific proteins whose genes had been identified through genetic studies. This rational approach to drug design accelerated the development of new therapies. Vaccines, too, benefited from molecular understanding. Traditional vaccine development often required growing viruses in eggs or cell cultures, then inactivating them. DNA technology enabled new approaches, including producing just the specific viral proteins needed to trigger immune responses without requiring the whole virus. The speed and precision of these techniques would prove particularly valuable when rapid vaccine development became necessary. Gene therapy represented perhaps the most direct medical application of DNA knowledge. If diseases result from defective genes, could introducing functional copies of those genes treat the disease? Early attempts faced significant challenges. Delivering genes to the right cells, ensuring appropriate expression levels, and avoiding immune responses proved more difficult than initially hoped. Some early trials encountered serious complications, sobering the field's initial optimism. Yet persistence and improved techniques gradually brought success. Treatments for certain inherited forms of blindness, where defective genes in retinal cells cause progressive vision loss, showed that delivering functional genes could restore some vision. Therapies for severe combined immunodeficiency, a condition where children are born without functioning immune systems, demonstrated that gene therapy could provide lasting benefit. These successes came after decades of setbacks, refinements, and careful work to understand what had gone wrong in earlier attempts. The production of monoclonal antibodies, proteins that bind to specific targets with exquisite precision, became another major application. These antibodies serve as targeted therapies for conditions from cancer to autoimmune diseases, Creating them requires understanding the DNA sequences that encode antibody proteins and the ability to manipulate cells to produce desired antibodies in quantity. As DNA technology matured, questions emerged alongside capabilities. Access to genetic information raised considerations about privacy, discrimination, and the psychological impact of knowing one's genetic risks. The technical capacity to read and use genetic information advanced faster than society's frameworks for addressing these questions. The power contained in understanding DNA's four-letter code brought both promise and responsibility. Through all these applications, the fundamental discovery remained the same elegant double helix that Watson and Crick had modeled decades earlier. Those four chemical letters, arranged in specific sequences, contained information that could now be read, interpreted, and applied to heal and help. The abstract beauty of DNA's structure had become tangibly useful in ways that touched millions of lives, transforming the story of DNA from pure science into practical medicine. As the 21st century dawned, scientists found themselves holding something remarkable in their hands. After decades of learning to read DNA, they were now learning to write it. The ability to edit the genetic code with precision had moved from the realm of science fiction into laboratory reality, opening doors that previous generations could scarcely have imagined. The story of genetic editing begins not in a research laboratory studying human cells, but in the microscopic world of bacteria. These single-celled organisms had been fighting their own battles against viruses for billions of years, and in doing so, they had evolved an ingenious defense system. Scientists studying bacteria in the early 2000s noticed something peculiar in their genomes. There were strange repeating sequences of DNA, with unique sequences sandwiched between the repeats. These patterns had been observed as early as 1987, but their purpose remained mysterious. The sequences were given an awkward but memorable name, CRISPR, which stood for Clustered Regularly Interspaced Short Palindromic Repeats. As researchers investigated these curious genetic patterns, they discovered something extraordinary. The unique sequences between the repeats were actually snippets of viral DNA, like wanted posters kept on file by the bacterial cell. When a virus attacked, the bacterium could use these stored sequences to recognize the invader and mount a defense. The defense mechanism itself was equally fascinating. Working alongside the CRISPR sequences were genes that coded for proteins called CAs, or CRISPR-associated proteins. Together, this system functioned like molecular scissors guided by a genetic memory. The bacterium would produce RNA copies of the stored viral sequences, and these would guide the CA's proteins to find and cut any matching DNA. It was a sophisticated immune system operating at the molecular level. Scientists Francisco Mojica in Spain, Rodolphe Berengu in the United States, and others pieced together how this bacterial defense system worked through careful observation and experimentation. But the breakthrough that would transform biology came when researchers realized this system could be repurposed as a precise tool for editing any DNA sequence. Jennifer Doudna at the University of California, Berkeley, and Emmanuel Charpentier, then working in Sweden, published landmark research in 2012 showing how the CRISPR-Cas9 system could be programmed to cut DNA at specific locations. The elegance of the system was stunning in its simplicity. Scientists could create a short piece of guide RNA that matched any DNA sequence they wanted to target. This guide RNA would lead the Cas9 protein, which acted as molecular scissors, directly to that spot in the genome. Once there, Cas9 would cut both strands of the DNA double helix. The cell's own repair machinery would then kick in, either disrupting the gene at that location, or, if provided with a template, inserting new genetic information. What made CRISPR-Cas9 revolutionary was not just its precision, but its accessibility. Previous methods for editing genes were laborious, expensive, and often imprecise. CRISPR was faster, cheaper, and far more accurate. Laboratories around the world quickly adopted the technology, and the pace of genetic research accelerated dramatically. Researchers began using CRISPR to edit genes in bacteria, plants, animals, and human cells grown in laboratory dishes. They could now turn genes on or off to study their function, correct mutations that cause disease, or introduce new genetic instructions. The tool proved remarkably versatile. Scientists edited the genes of mosquitoes to prevent them from transmitting malaria, modified crops to resist disease, and created laboratory models of human genetic disorders to better understand and potentially treat them. The technology also revealed how much scientists still had to learn. Sometimes the Cas9 scissors would cut at unintended locations in the genome, creating off-target effects that needed to be carefully monitored and minimized. Researchers worked to refine the system, developing new variants of CA's proteins with greater precision and new ways of delivering the editing machinery into cells. As CRISPR technology matured, discussions began about its potential therapeutic applications. Could genetic editing be used to treat human diseases caused by faulty DNA sequences? Early clinical trials explored using CRISPR to modify immune cells removed from patients, editing them to better fight cancer or HIV, then returning them to the body. These approaches focused on editing cells outside the body, where scientists could verify the changes before the cells were reintroduced. More ambitious possibilities also emerged. Researchers investigated whether genetic editing could be performed directly inside the human body to correct disease-causing mutations in conditions like sickle cell disease or inherited blindness. Some of these inherited conditions result from errors in a single gene, and correcting that specific sequence could potentially address the root cause of the illness. The ability to edit DNA also prompted deep reflection about how such powerful technology should be used. The scientists, ethicists, and policymakers engaged in thoughtful discussions about appropriate boundaries and guidelines. Questions arose about the difference between editing cells in an adult patient, where changes would affect only that individual, versus editing embryos or reproductive cells, where changes could be inherited by future generations. These conversations became urgently relevant when a researcher announced in 2018 that he had edited the genes of human embryos that were then brought to term, resulting in the birth of twin girls. The international scientific community responded with widespread criticism of this premature and ethically questionable application of the technology. The incident underscored the importance of careful deliberation about when and how genetic editing should be applied, particularly in ways that would affect future generations. Meanwhile, researchers continued developing the technology, discovering new CA's proteins with different capabilities and refining the delivery methods to make editing more efficient and precise. Scientists found proteins that could edit single letters of the genetic code without cutting both strands of DNA, opening up even more nuanced possibilities for genetic modification. The tools for rewriting the code of life were becoming more sophisticated with each passing year. As scientists gained the ability to edit DNA with increasing precision, they found themselves stewards of a profound capability. The same double helix that Watson and Crick had modeled with metal plates and wires in Cambridge 7 decades earlier could now be deliberately rewritten, letter by letter. The story of DNA had entered a new chapter, one where humanity was learning not just to read life's instruction manual, but to carefully edit its pages. The tools for reading and editing DNA have opened windows into questions that once seemed forever beyond our reach. As we settle into this understanding, we find ourselves able to look backward through time with remarkable clarity. The DNA molecule, it turns out, is not just a blueprint for building living things in the present moment. It is also a historical document, a record written in 4 chemical letters that preserves the story of life across billions of years. When scientists compare DNA sequences from different species, patterns emerge like constellations in the night sky. The genetic code of a human being shares remarkable similarities with that of a chimpanzee, somewhat less with a mouse, still less with a chicken, and yet even a banana carries genes that bear recognizable kinship to our own. This nested pattern of similarity and difference reveals the branching tree of life, showing how all living things descend from common ancestors in the distant past. The molecular clock ticks steadily in these sequences. As generations pass, random changes accumulate in DNA at roughly predictable rates. By counting the differences between the genetic sequences of 2 species, scientists can estimate when their evolutionary paths diverged. The technique has allowed researchers to construct timelines reaching back through the ages, tracing lineages and relationships that no fossil record could fully illuminate. DNA extracted from fossils and preserved remains has opened particularly fascinating windows into the past. In laboratories around the world, scientists have recovered fragments of genetic material from bones tens of thousands of years old. The DNA of Neanderthals, our extinct cousins, has been sequenced and compared with modern human genomes. The comparison revealed something unexpected and intimate. Many people today carry small percentages of Neanderthal DNA in their genomes, evidence of ancient encounters and interbreeding between our ancestors and theirs. The past is not separate from us, but woven into the very fabric of our cells. Similarly, DNA analysis has revealed another group of ancient humans previously unknown to science, the Denisovans, identified initially from nothing more than genetic material extracted from a small finger bone found in a cave in Siberia. Their DNA too persists in some modern populations, particularly among people in Oceania. The genetic record preserves traces of human relatives and human journeys that left no written documents, no monuments, only the quiet testimony of molecular sequences passed down through countless generations. The technique of sequencing ancient DNA has extended beyond humans. Scientists have read genetic information from woolly mammoths frozen in permafrost, from extinct ground sloths preserved in dry caves, and from plant seeds thousands of years old. Each sequence offers glimpses into vanished ecosystems and evolutionary experiments that time has closed. Yet DNA is not merely a record of the past. It continues to evolve in the present, changing slowly through mutation and natural selection, responding to environmental pressures and random chance. In laboratories and natural habitats, scientists observe evolution occurring in real time. Bacteria develop resistance to antibiotics through genetic changes that spread rapidly through populations. Viruses mutate as they replicate, their genetic sequences shifting subtly with each generation. The process that Darwin described without knowing its mechanism continues all around us. Written in the language of nucleotides, the unity revealed by DNA is perhaps its most profound message. Despite the tremendous diversity of life, from microscopic bacteria to towering sequoia trees, from deep-sea tube worms to mountain eagles, all living things use the same genetic code. The codon that specifies the amino acid leucine in a human cell specifies leucine in a bacterium as well. This universal language suggests that all life on Earth descended from a single common ancestor. Billions of years ago, some early cell that stumbled upon this four-letter alphabet and passed it down through every subsequent generation. This molecular unity has practical implications. Because the genetic code is universal, scientists can transfer genes between vastly different organisms. A gene from a jellyfish that produces a glowing green protein can be inserted into the DNA of a mouse, and that mouse's cells will faithfully read the jellyfish gene and produce the glowing protein. This technique has become an invaluable tool for research, allowing scientists to track the activity of specific genes and observe biological processes that would otherwise remain invisible. The sequencing of thousands of genomes from diverse species has revealed unexpected evolutionary relationships. Some organisms that appear similar turn out to be only distantly related, their resemblance the result of convergent evolution, solving similar problems with similar solutions. Others that look quite different share deep genetic kinship. The whale swimming in the ocean carries in its DNA the unmistakable signature of its descent from land-dwelling mammals, its flipper bones, arranged in the same pattern as the bones in a human hand. As technology improves, the ease and speed of DNA sequencing continue to increase, while costs decrease. What once required years of effort by large teams can now be accomplished in hours by automated machines. This democratization of genetic technology has spread DNA analysis into fields far beyond basic research. Conservation biologists use DNA to track endangered species and assess genetic diversity in small populations. Forensic scientists read DNA sequences to identify individuals and solve crimes. Doctors increasingly incorporate genetic information into medical decisions. The story of DNA has moved from a time when heredity was mysterious, through the careful observations of monks and microscopists, past the elegant discovery of the double helix, and into an era where reading and even editing genetic code has become routine. Yet each answer generates new questions. How do regulatory sequences control when and where genes are active? How do organisms with relatively few genes create such complexity? How do genomes evolve new capabilities? The four chemical letters adenine, thymine, guanine, and cytosine continue to spell out messages we are only beginning to fully comprehend. In the quiet spiral of the double helix resides not just the instructions for building a single organism, but the accumulated wisdom of billions of years of life, testing and refining solutions to the challenges of existence. The story of DNA is ultimately the story of life itself, written in the most fundamental alphabet of biology, connecting every living thing in an unbroken chain, stretching back to the very beginning and forward into an open future. As we bring to a close our journey through the elegant double helix and the brilliant minds who unveiled life's secret code, we hope this story of discovery has gently carried you toward rest. From Watson and Crick to Franklin's crucial insights, we've explored how four simple letters write the story of every living thing. Thank you for joining us on this peaceful exploration. If these calm adventures have brought you comfort, we'd love to have you with us again. Like, subscribe, and share your thoughts in the comments below to help other smart sleepers find their way here. And now, as the night deepens and your mind settles, we wish you the sweetest of dreams. Good night, fellow dreamer, and may your sleep be restful and your dreams be kind.